Good. So good morning, everyone. Welcome to our sixth annual Participant Appreciation Day. This is for our research participants. My name is Victor Henderson, and before we get started, Dr. Nusha Skara will have some announcements for us.
A little louder. Yeah. Hold the microphone closer to carry that.
We will have some announcements.
Good morning, everyone, and welcome to our sixth annual Participant Appreciation Day. So happy to have you all here with us today in person. I know you might have friends who inquired about, can we do this, virtually, online, etc. So I am putting in a very big good word that we'll do this hybrid next time, but we would like you all to try and come back in person for sure. Okay, this is so important for all of us to be together as a big happy family. So we really appreciate your being here today, and we appreciate your spirit of generosity and volunteerism throughout the Healthy Brain Aging study. So I have a few little house guidelines for you all. Kindly first, make sure that your cell phones are on silent. Secondly, I think an important thing would be to tell you where the restrooms are. So if you go out through those double doors, when you face the elevator, you make a slight left and turn around there, and you can go that way. Now, I know there are a limited number of restrooms on this floor, so there are a few people. One of them is Nora. She's wearing a greenish teal shirt. She's outside at the sign-in. She has a badge to help people go upstairs to use a restroom. And then Alyssa and Jamie, they're waving their hands in the back. Please see one of them if you need restroom access. I'd also like to acknowledge Jamie and Alyssa's help today with helping getting this event together. So thank you both so very much for your kind help. Likewise, we have Christina Weiss-Corey, Nora Sakis, Nicole Caceres, Hannah Schmidt, and Leah Varghese from the Clinical Core helping out with the logistics of sign-in and greeting all of you. So I wanted to also acknowledge them. They're still outside, but let's give them a round of applause as well. Thank you. So the other logistics are this. Every speaker will give their presentation. We've allowed maybe about 1 or 2 minutes at the end if there's a need for a clarification only. So if there was something in their slides or their presentation you didn't understand, please raise your hand and we'll try and get a microphone to you to ask the speaker. They will call on you directly. The Q&A session is scheduled and it's on the agenda. It'll be after the sorry, I'm trying to see where it is. Yes, 11:50. So it'll be after the presentation by Dr. Schule. So we have given you all on your cards, on your desk tables there are index cards with pens. You could please write your questions as they come up. You don't have to wait till the Q&A, and we actually ask that as they come up, you do it, and then you can just gently hold it up, and somebody will come around and take it from you. They will collate those index cards, try to consolidate things as needed so that you can we can have the Q&A move efficiently that way. So they will read the question to the speaker. The speaker will then, please make sure you write the question and which speaker it is intended for. Very important. Who is this question being addressed to? Yes. Okay. I'm sitting right here. Feel free to nudge me. I'm happy to come over and take your question. Okay?
And if you can't remember speakers' names, that's okay. We'll sort of match the question to the speaker as best we can.
Well, yes, but I guess I want to make sure we take it the way it's intended, but sure, that would be the other option. Lastly, in the afternoon, we will have some breakout sessions. Dr. Goldman Rosas will be here later as she introduces the Community Advisory Board, and they'll have some breakout sessions, and she'll give you guidelines around that as well. At the very end then, because I know some people said they are leaving earlier, we have added in your agenda that I emailed to all of you. And by the way, I'm sorry, I should have said, I'm Nusha. I'm the face behind the ADRC emails that you get all the time. So it's me. Kindly take that survey and give us your constructive feedback so that we can keep trying to do our best to improve this event for all of you. Okay? Are there any questions here? Thank you so much. Then, without further ado, I'll pass the mic back to Dr. Henderson.
Hey, good morning, everyone. And thanks again for being here. I heard there's rain coming tomorrow, but today is a good day. So thank you for that. And there's plenty of coffee in the back. So my name is Victor Henderson, and I am the one responsible for trying to organize as much as I can. And we all do the best we can. Nusha helps out tremendously in this regard. This is our sixth annual Research Participant Appreciation Day. It's a conference on healthy brain aging. It involves a lot of people behind the scenes, and a lot of people volunteering for the research that we do here. So we're representing primarily, but certainly not exclusively, the Healthy Brain Aging Study, under whose aegis the Alzheimer's Disease Research Center exists. But also there are a number of other groups that are full partners here. The Lewy Body Scientific Partnership for Advancing Research and Knowledge. This is a new acronym for the old UDAL cohort from representing Parkinson's disease, an important part of what we're looking at within the Alzheimer's Disease Research Center. There are clinical trials programs at Stanford that are partners with us in a lot of different ways. There is a Stanford Aging and Memory study. There's the LEAD study, the Longitudinal Early Onset Alzheimer's Disease study. We have participants from that research. And the newest kid in the block is the Asian cohort for Alzheimer's disease. So all these are sort of encompassed and related ongoing overlapping research projects. And we thank everyone for helping out in these various projects that we have. The ADRC includes a lot of people. I think this is most of them, but this includes faculty who are involved, staff members, research coordinators. It includes some of our postdoctoral fellows and people who have received are receiving ongoing grant support from us. So a lot of people involved in this organization. I'll give another shout out to Nusha Skari, to Elisa Anderson and Jamie Kramer, who helped organize today's session, and also helping out today, Christina Weiss-Correa, Leah Verghese, Nicole Caceres, Hannah Schmidt, and Noor Sakids also are very much appreciated. Their help is appreciated in making today possible. We're also being joined today by some of the members of our community advisory boards. We refer to this as our CAB. This is a group that Lisa Goldman Rosas has helped organize to advise our center on different aspects of the community, how to engage the community more fully. And you'll be introduced to some of these people later during this program, but I'll leave that for later on. This center has been around since 2015, and it's been quite productive. I just went on to the National Library of Medicine website and tried to see how many research publications have cited the grant from our particular center. So it's about 100 publications every year that result from what you, the participants, do in terms of the clinical data that you contribute, those of you that have had brain imaging done, who have donated blood, stool microbiome, spinal fluid, DNA, actigraphy, sleep measures, just a whole host of measures. And these are used by investigators here at Stanford. They're also shared, some of these, with national databases in an anonymized way to help researchers at other sites. And so not all of these publications, in fact, probably not the majority of these publications, are primarily based just on Stanford research, but represent shared data across the different Alzheimer's disease research centers and other cohorts and consortia of researchers interested in neurodegenerative disorders like Alzheimer's disease, like Parkinson's disease, and dementia with Lewy bodies, and related problems of aging and cognitive impairment during aging. Oops. So in terms of what we're going to be talking about today, the first part of the program, and a lot of the program, is to tell people more about what actually goes on in the center, what happens to some of the data, some of the images, some of the fluids that you contribute here. And so we'll have presentations, for example, from Dr. Mike Gracious about some of the ways that genetics are being used to advance Alzheimer's disease. And we'll hear about some of the studies, just one small part of them, based on blood-based biomarkers from Tony Weiss-Correa about aging and about different disease processes. Dr. Birgit Schuler will tell us why we're asking for skin samples, little tiny skin biopsies, but they can be used for so many important things related to disorders like Alzheimer's disease, like Parkinson's disease, and we'll hear about some of the work that she's doing. There'll be a lunch break, and then after that, we'll hear about some other findings. Dr. Ryan Taylor will tell us about the lumbar puncture. Some of you have been able and willing to contribute spinal fluid, which is really quite valuable and a resource for a lot of the research that's being done here and elsewhere. We're very fortunate to have really an award-winning journalist, Selena Rodriguez, who is here. She's also a research volunteer. She's a CNN journalist, and she's had a lumbar puncture, and she will let us know how it went for her. Hopefully, okay. Well, I'm hearing snickers, but I'm also seeing the thumbs up. So I think we will be okay. We'll hear something about justice, equity, diversity, and inclusion. This shows up in the news in a lot of different contexts in a lot of different ways, but there are important scientific issues involved. And so there will be a presentation from Dr. Patricia Rodriguez Espinosa. We'll hear more about our wonderful new community advisory board from Dr. Lisa Goman Rosas, who will then lead us in breakout sessions related about what the feedback that you have from us, and she's entitled that we want to hear from you. Now I'm going to lead off, and I'm going to lead off with something that's actually not coming directly from our center, and that is looking at some of these new treatments that have been approved for the treatment of Alzheimer's disease. This has gotten a lot of attention in the news, a lot of attention within the scientific world, a little bit of controversy, a lot of a lot being said. And because this is something that comes up a lot and a lot of people have questions about it, I thought I would give an overview on at least what my perspective is for these new drugs. So what you'll see when I talk about these new approved treatments for mild cognitive impairment and mild dementia due to Alzheimer's disease is I sort of view this as a glass half empty, a glass half full. So there's a lot of promise, a lot of enthusiasm, but also some caveats, and I'll try to go into those as best I can. As many of you know, that before in 2021, there were several approved therapies for dementia due to Alzheimer's disease. These are drugs like donepezil or rivastigmine or memantine that some of you have heard of and are more familiar with, but there were no approved therapies for the treatment of mild or cognitive impairment. But that situation has changed. And so there are now FDA-approved treatments for both mild cognitive impairment as well as for mild dementia associated with an Alzheimer's disease biomarker. And I'll explain more about what that biomarker is, but essentially it's referring to amyloid image on PET brain scans. These new drugs are all antibodies that target beta amyloid. That's one of the abnormal misfolded proteins within the brains of people who have Alzheimer's disease. And the three drugs, and I'll talk about each of these. One of these is aducanumab, which received a very controversial accelerated approval by the FDA in 2021. The other is lecanemab, which was approved just several months ago by the FDA, full approval. And the third one is donanemab, which we expect to receive full approval by the FDA later this year. And these drugs all work again on removing amyloid. So one of the histological biochemical hallmarks of Alzheimer's disease is the amyloid plaque, something that neuropathologists can see under the microscope. This is a depiction of a plaque with amyloid staining, and these occur in the brain between nerve cell bodies in certain regions of the brain. And these were described originally by Alois Alzheimer and his first patient, Augusta D, in publications in 1906, 1907, 1911. And he described the two key microscopic features of Alzheimer's disease. The first were these amyloid plaques, and these are Alzheimer's drawings of what he thought the plaques looked like. And you can see that he got a lot of it right in these original microscopic descriptions. And the other change he also described that was seen in the brain of Augusta D were these nerve fibrillary tangles. These are inside nerve cell bodies, not between nerve cell bodies. These are composed of a different important protein, tau protein, that has too many phosphate groups on it. It's hyperphosphorylated. It's also misfolded. These are more modern pictures of what these tangles look like, but you can also see the resemblance to Alzheimer's original drawings. So there's a putative sequence by which pathology occurs in Alzheimer's disease. This is more a hypothesis than an established fact, but it's a very strong hypothesis, and that is the earliest biochemical changes in Alzheimer's disease is this amyloid, amyloid being deposited and forming eventually these plaques that can be observed in the brain. It's thought in some way to trigger the hyperphosphorylation of tau protein. These accumulate as tangles within vulnerable neurons. Over time, this will damage the nerve cells that will lead to a loss of synapses or connections between nerve cells, a loss of communication among different parts of the brain, ultimately death of nerve cells. Sometimes this can be detected by shrinkage on MRI brain scan or CT brain scan. And eventually, and only at a later stage, does cognitive impairment occur, where memories affected other aspects of cognition, first in a mild form and then later in a more severe form represented by dementia. So this is a long sequence, probably taking several decades, and based on newer studies coming out of a lot of Alzheimer's centers, including imaging studies done here, it's now known that about a third of cognitively normal people aged 70 or older will have amyloid in the brain, as demonstrated by some of these new techniques. And so amyloid accumulation occurs pretty early on. Given this hypothesis that amyloid occurs early, it's important in some of these other sequences that occur. It's not surprising that a lot of the drug therapy research is focused on ways of trying to reduce amyloid in the brain. And this was given an important impetus in 2016. Thank you. The small print looks smaller and smaller these days. So this was sort of a smaller, what's called a phase 1 trial, looking at the first of those 3 drugs I talked about, aducanumab, given by intravenous infusion over a 12-month period of time. We're looking at images from amyloid PET scans of the brain, looking at placebo at the top, and then sort of lower, middle, and higher doses at the bottom, before and after slides. And you can see, as suggested by these, that a lot of amyloid is being removed from the brain. So this is exciting, and so I'll give this a big red, exciting check mark for that. And this was replicated. It was replicated in 2 large phase 3 clinical trials involving about 1,800 people in each of these over an 18-month period of time. And you might be able to see at the top of the slide that the placebo group showed no reduction in amyloid. None would be expected, of course. And the higher dose aducanumab showed big reductions. So a big red arrow there, another big red arrow there. Now, trying to see what the clinical effect is for these treatments, there are different ways of doing this. A common way is based on something referred to as a clinical dementia rating, sum of boxes scores. It sounds complicated. It's actually pretty simplistic. It's a structured interview that rates people on memory, orientation, judgment and problem solving, community affairs, home and hobbies, and personal care. It ranges scores for each of these range from 0 to 3. And so if you do the multiplication, that means the potential range of scores adding up these boxes is 0 to 18. And it's thought that the minimal difference that would be clinically discernible, clinically meaningful, might be 1 or 2 points in this 18-point scale. And this was a primary outcome that was used in the aducanumab clinical trials, and it's a common outcome in other trials as well. And the message here is a little bit more mixed. There were 2 large phase 3 trials, and the first one did show a difference. It showed a difference in the group that received the active treatment, the aducanumab, and it was statistically significant. But the overall difference was 2/5 of a point, so less than the 1 or 2 point minimal clinically important difference. But we'll give that a little check. So it seemed important, but not quite meriting the large red check that was given before. The other trial didn't even show that. There was no difference. And it was this discrepancy that confused the FDA. There was an important effect on a biomarker, the PET scan. It's sort of a small discrepant result in a clinical outcome. And so they used an unusual approval mechanism, accelerated approval, for this drug. So the second drug, the one that has full approval that's available now, is lecanemab. This was also tested in a number of trials, including a large phase 3 clinical trial where people received either placebo infusions or lecanemab infusions every 2 weeks for an 18-month period of time. These are people who had either mild cognitive impairment with evidence of amyloid from PET scans before at the start of the trial, or people with mild dementia due to Alzheimer's disease. And the primary outcome here again was the clinical dementia rating sum of boxes. So looking at the amyloid results, huge reductions. These drugs work. They remove amyloid from the brain, a big red check mark. In terms of clinical outcomes, let me make a couple points here. So there was a difference at the end of 18 months. The difference favored people who received the lecanemab, but the difference was small. It was now almost a half a point between the two treatment groups. I'd also point out that average scores got worse in both treatment groups, both placebo and the lecanemab group. There was a statistically significant difference in the end of 18 months, but that's still in the little check category. But there is something else, and that is if you look at the rate of getting worse, then the rate of getting worse was about 27% better in the active treatment group. I'm not sure exactly that serves a big red arrow, but it gets sort of slightly larger, but still smallish arrow. The third drug, the one we expect to receive full approval later this year, is donanemab. It's also given by intravenous infusion. It was also tested in a large phase 3 trial over an 18-month period of time. The primary outcome here was a different measure, but they also looked at the CDR sum of boxes score. And here, on average, people in both groups got worse, but the difference at the end of 18 months was 2/3 of a point. So it's sort of inching upwards, but it's still probably not a detectable difference, but it was statistically different. For the primary outcome, it's something called the Integrated Alzheimer's Disease Rating Scale. There was a 3-point difference, but this is a 144-point scale. And the 3 points was also not considered to be to reach a threshold of a minimal clinically important difference. And so this also gets sort of a smaller check mark. Looking at the rate of getting worse, the difference between the groups was a little bit larger. It was so about a 35% difference. So looking more interesting from that, looking at the numbers in that way. So these drugs, like all drugs, have side effects. The major side effect that people are concerned about is something referred to as ARIA, amyloid-related imaging abnormality, either related to edema or related to hemorrhage, so ARIA-E or ARIA-H. And this is because amyloid is found not only in the brains or people who have early Alzheimer's changes or Alzheimer's dementia, but also in the brain parenchyma and also within blood vessel walls often. And so as amyloid comes out of blood vessel walls, there can be leakage. This can show up as inflammation, edema, and there can be hemorrhages, often very small, but sometimes larger. And so the slide on the left shows an example of what this edema might look like on an MRI scan. Sometimes this is asymptomatic. Sometimes it causes headache, confusion, rarely seizures. Hemorrhages are often tiny. They're little tiny black circles. In the second slide would be examples of microhemorrhages. These may be not noticeable by patients. There can be evidence of prior bleeding. That's the superficial siderosis referred to here in one of the slides. And occasionally, fortunately not commonly, there can be very large hemorrhages as well related to amyloid in blood vessels and these treatments. So how often does this occur? This is a fairly busy slide, but I'm showing drug placebo differences in the different phase 3 drug trials looking at ARIA-E, and drug placebo differences range from about 11% to 33%. Looking at microhemorrhages, the range is from about 6% to 14% placebo versus people in the active treatment groups. Again, some of these are asymptomatic. And there are also infusion reactions that can occur with these drugs given intravenously. This can be hypertension, hypotension, nausea, vomiting. There's some others as well. And you can get from this the impression that there might be an opportunity for an unblinding. So even though people don't really know for sure whether they're getting an active treatment or placebo, having even an asymptomatic edema showing up on an MRI scan, which was monitored during the course of the trial. This leads to a change in protocol with more frequent monitoring and so forth. And some of the infusion reactions may also be symptomatic. And ordinarily in a randomized, double-blind, placebo-controlled trial, we wouldn't worry about these little differences. But when the differences are very tiny, then it's hard to know whether the small differences that we're seeing are really related to the intended effect of these drugs or maybe to some other effect. We just don't know. And finally, there's sort of this interesting observation that there's some loss of brain volume that's associated with treatment. There's some brain shrinkage in everyone. The amount of brain shrinkage is fairly small. But it seems to be larger than could be accounted for just by the removal of the amyloid from the brain. We don't know what the long-term significance of any of this might be, but it's just something else that investigators need to keep in mind. So there are other considerations. These drugs are fairly expensive. Lecanemab, for example, is being priced at about $26,000 a year, but that's really only a small part of the price because there are prices involved in a lot of the screening, the infusion center costs, the need for, I think, 4 MRI scans as part of routine monitoring, the costs involved in looking at symptoms that might represent ARIA and so forth. So the true cost of these drugs are probably $100,000 or $200,000 per year. And that means that there are a lot of resources that are needed for screening, for treatment, and for monitoring for safety purposes. And that brings up an issue of equity. The people who are from poor or more disadvantaged groups may not have access to the specialized treatment. In terms of monitoring, some of the side effects may be picked up less readily. These are people who sometimes are at greater risk for developing dementia in the first place. And so these are important issues I think everyone is thinking about these days. So is the glass half empty or half full? I think I'll answer yes to both of those. And my concluding thoughts are that it is exciting that for the first time there are approved treatments for not only Alzheimer's disease, but also mild cognitive impairment associated with an Alzheimer's biomarker. This has certainly energized the field. It certainly energized pharmaceutical companies who know that there are billions to be made. And I think will spur efforts to find something that's even better than what we have now. I think we need to keep in mind that the patients worsen despite treatment for these drugs. The average difference in decline at 18 months seems important. For lecanemab, the rate of getting worse was 27% better. But the average clinical difference at 18 months is still probably too small to be noticeable. What we don't know is what the long-term clinical outcomes are. It may be that these outcomes are much better 5, 7, 10 years later, or they may not be. We don't know. There's more research that's needed to show that. There may be long-term clinical benefit. There may be long-term harm related to some of these accumulating side effects and other things we don't know as much about yet, or there may or may not be much of a difference. So there are now about 33 Alzheimer's disease research centers scattered across the country. Some of them, like ours, look at neurodegenerative disorders like Parkinson's disease and dementia with Lewy bodies. Others have other kinds of things that they focus on. Most of the centers don't primarily do drug treatment trials, and ours is one of those centers where our focus is a little bit different. I think all of these centers are contributing to fundamental advances in understanding the diseases, trying to understand better what approaches might work, what preventative approaches might work. But there are also people doing clinical trials. There's a clinical trials group here within the memory disorder division in the neurology department, also within the movement disorders division. And with that, I will thank you for your attention. And if there's time later for questions or during the break, I'll be glad to answer those. Thank you very much. I see two. So pink shirt and then red. So go ahead. I saw your hand first.
Thank you. Just very quickly, I looked up siderosis, and it said something about it's the result of iron deposits
Yes.
of iron something or another. But what's the significance of siderosis?
So when I showed the MRI scans of some of the side effects, one of the hemorrhage side effects of ARIA was superficial siderosis. So siderosis, as you say, refers to iron. And you might remember, iron poor blood. So red blood cells, the hemoglobin carrying capacity involves an iron atom. And so when there's been bleeding, the iron can be left behind. It can layer out over the folds of the brain. And with certain MRI sequences, this can be visualized quite readily. And so it's just evidence that there's been some prior bleeding there.
Yes, hello. I've been involved with this study since the beginning.
Thank
and
you.
I wondered then, as now, are there efforts, as I look around this room, I do see a predominance of Caucasian people. And I noticed you had the Asian consort, was that the term?
Well, cohort, it's just a
Are
fancy.
efforts being made to be sure that we are inclusive in terms of ethnic backgrounds and race, et cetera?
So that's a large we that you're referring to. And I think the NIA is very cognizant of this. Some of these studies are government industry partnerships, some aren't. And I think industry is also aware that the NIA cares and therefore they should care. And so in reporting the results from lecanemab, reporting the results from donanemab, there was an effort to sort of say who was, what groups were included. That said, the people who primarily volunteer for research, like me, not everyone in this room, but many people tend to be overeducated, privileged. People who know the value of research, aren't too ill with other things, have the resources to make it to a research setting. And so I think people who are at higher risk are underrepresented in clinical trials. And I think everyone is trying to do a better job, not always for the right reason, but often for the right reason. So I think we're getting there, but it's still not very good. Thank you so much. So our next speaker is Dr. Michael Gracious, who has been with the ADRC from the very beginning. He is the associate director, co-director going forward in our renewal application coming up in a year. So this will be a big year for us as we try to put everything together. He has had a major role in the imaging core, and some of his research in the last several years has transitioned to looking at genetics in relation to aging, relation to neurodegenerative disorders. And his approach in some ways is sort of far ahead of what a lot of other groups are doing.
All right. Thank you, Victor. Thanks, everybody, for coming. I have to say this is one of my favorite audiences to speak to. I'm glad you guys are all here. I've made an effort to, teach everybody here genetics in 10 minutes, which might be a little ambitious, but we'll walk you through some important points. And mostly I wanted to sort of emphasize the sort of stuff that we're doing here at Stanford that really can't be done elsewhere as yet. We're kind of leading the charge on this. And this is genuinely and truly thanks to you guys because of the special genetics that we're doing on your samples that you've donated. All right, so a quick primer on the genetics of Alzheimer's disease. We kind of break it up into two parts. One are the genes that are involved when people get sick very early with Alzheimer's disease. So these tend to run very strongly in families. They're called autosomal dominant. And just one mutation on one of these genes can get passed on across generations and cause Alzheimer's disease in anybody and everybody actually that carries a copy of these genes. These are very rare. So less than 1% of the people that we see in clinic will end up having one of these variants, but we have several families in the ADRC that carry some of these variants, and it's an incredibly powerful research tool. As devastating as it can be clinically, it's also an incredibly powerful way to think about Alzheimer's disease because we know these people are bound to get sick. And these are the three genes that you don't need to memorize on which mutations can cause these early onset Alzheimer's disease cases. The main thing to know about these is that they all point very directly to amyloid. So one of the genes is amyloid precursor protein. These two other genes, presenilin 1 and presenilin 2, actually cut amyloid precursor protein. So all the very early strong genetics points to amyloid, which is part of why we focus so strongly on amyloid in our treatment approaches. But most of what we think about in the clinic is sort of later onset. People get sick in their 60s and 70s and 80s. And here, the main gene that stands out that you may have heard of is apolipoprotein E, or ApoE. And in particular, this specific variant, which is very common, about 15% of people in this room have one copy of ApoE4. E4 increases risk for Alzheimer's disease, but doesn't guarantee that you're going to get it. Plenty of people live into their 90s, die without dementia with one and even two E4 variants. But it's a strong risk factor. And then there are a bunch of other genes, variants on genes that are not as strong as ApoE4, but that are important for helping us think about different pathways that we might target with drugs. Even if they're not any one of these other variants isn't a powerful predictor, it can help us think about the disease mechanisms. Okay, so I'm going to walk you through what this is called a Manhattan plot for maybe obvious reasons. This is supposed to look like a skyline. And along the x-axis here, you see 22 chromosomes labeled 1 through 22. And basically every little column here is a single genetic variant. And for the most part, very common variant. So this particular variant, maybe 20% of people have the reference, or sorry, 80% of people have the reference, and 20% of people will have the alternative nucleotide. So there are 4 nucleotides, A, T, G, and C. And if you switch a T to an A, we call that a variant. And some of those variants can increase risk for Alzheimer's disease. So every column here is a genetic variant, mostly common. The y-axis tells you how significant that variant is in terms of its Alzheimer's risk. So the higher up on the y-axis, the stronger the genetic risk. And so when you look at this, I've been talking about ApoE, you might say, well, hey, what's happening here with BIN1, this other variant? Looks like it's also very statistically significant. And it is, but what happens with ApoE is so strong that they actually have to kind of truncate the y-axis. So if we were to see the true statistical strength of ApoE, it would be 880 on this y-axis. So it'd be on the 4th floor of the building or something. So ApoE really dominates the skyline here, but these other variants are important. A lot of these, about 5 or 6 of these variants point to a cell called microglia, which we think is really important in Alzheimer's disease. And that interest in microglia was largely based on these genetic studies. So even if they aren't powerful predictors, any one of these, they really help us think about the disease process. Okay, so we can sort of divvy up these different genetic variants based on the strength of their effect on the y-axis, how powerful they are at increasing risk for Alzheimer's, versus on the x-axis, how common these genetic variants are. And when we do this, we put these 3 genes on which mutations cause early onset Alzheimer's disease way up in the upper left. So they have a huge effect size, high risk, but very rare. Then we can look at ApoE, which is, we're kind of lucky in Alzheimer's disease. A lot of disorders don't have a gene like ApoE, which is actually, or ApoE4, the variant is quite common, and the risk is actually quite strong. Not definitive like these other genes, but quite strong. And this can help us sort of leverage ApoE4 status to think about other variants that might protect people. So our group does a lot of this, looking for people that carry 2 copies, for example, of ApoE4, who might be in their 70s or 80s and are still doing great. And the question is, despite this gigantic risk of 2 copies of ApoE4, why are they still doing well? Can we find protective variants in people like that? All right, and then there are a bunch of other variants from that Manhattan plot that are very common also, but have a much smaller effect size. And these, I have pointed out, are more important for thinking about the pathogenic pathways. You might be interested in your ApoE genotype. You're definitely not going to bother figuring out your genotype for these other ones because they don't convey very much risk. All right, so that's your primer. Everybody understands genetics and Alzheimer's disease now, great. And now we're going to talk about, I think, more interesting things that are coming out of our ADRC, out of your blood and your DNA. Okay, so much of what we've talked about so far are these single nucleotide changes. You turn an A to a T, and that gives you some change in your risk. And single nucleotide variants or changes have been really powerful in helping us understand Alzheimer's disease. But we still probably only understand about 50 or 60% of the genetics of Alzheimer's. So there's a lot of what we call missing heritability. Right? We think there's more genetic influence, but we haven't found it yet. And one place that this missing heritability might exist are in what we call structural variants. So these are large, not single nucleotide changes, but say, 1,000 nucleotides that just get deleted from your genome. That's called a structural variant, a big deletion. And with standard short read sequencing, we can't really see those at all. those are kind of invisible to us. So here I'm going to just show you an example. This is not somebody from our ADRC, but someone that I saw in clinic who had a very rare disorder, but that we diagnosed ultimately at autopsy. And I can't sort of speak enough to the importance of autopsy, both for clinical purposes and for research purposes. But with the help of our neuropathologist, we figured out what this person had. I wasn't sure when I was following them in life. And this particular disease essentially always has a mutation on this gene called CSF1R. And we looked high and low. We did clinical genetics. We did research genetics. We couldn't find a mutation that explained this. And so this is this person's short read sequencing. The whole picture here is 1,000 nucleotides, right? So ATCG, there are 1,000 of these in this window that we're looking at. And each of the DNA fragments here then is about 150 nucleotides. So this is short read sequencing. And with that, we couldn't find any mutations on this gene. But one of the very first long read sequencing tests that we did was in this subject. So this is the same subject, short read sequencing. Here's their long read sequencing. And now here's the short read, these little 150 base pair fragments. Here, we've taken much larger fragments of this person's DNA. So some of these fragments run all the way across this 1,000 base pair window. And we can get fragments up to 10,000 or 20,000 bases in length. And what this allows us to do is pick up large structural variants. So not think about single nucleotide changes now, but this person has a gigantic deletion, something on the order of 300 base pairs, right on this gene that cuts off about half of this part of the gene. And so this is, unquestionably the cause of this person's disease, something we never would have seen on the short read sequencing, but that we can pick up with this long read sequencing. And the really exciting thing about your guys' help is that now we're doing long read sequencing on all the DNA samples that we acquire through participants in the Stanford ADRC. We started with short read sequencing, and that's helpful. So we have short read sequencing on pretty much everybody. We have long read sequencing on almost everybody now too. We're still sort of cobbling some missing participants together. And I'll just describe in this sort of last example, one early and I think important finding that's already come out of just looking at the 400 or 500 long read genomes from you guys. All right. So this is a bit of a sidebar. Frontotemporal dementia is a different kind of neurodegenerative disease. We see this in clinic. It's less common than Alzheimer's. But what was found is that there's a variant on this gene called, we'll just call it TMEM for simplicity, that's quite protective against frontotemporal dementia. And it turns out this exact same region of TMEM is also protective against Alzheimer's disease. So that's already interesting, right? It's protective against 2 neurodegenerative disorders. We'd love to figure out why. It's a lot stronger, the protection in frontotemporal dementia than in Alzheimer's disease. And this is what the gene looks like. These little green parts are the parts of the gene that get turned into a protein. We call those exons. And these 4 RS numbers are different genetic variants, all of which protect against frontotemporal dementia and Alzheimer's disease. And it's complicated because these 4 variants are always co-inherited. So if you have one variant, you have the other 3 variants, which makes it very hard to determine which of these variants is actually the protective variant, right? And that's what we care about from a biological and treatment standpoint. Okay, so it turns out, if you look at, again, this is now potentially from one of you in this room, ADRC data. This is the short read. You can see the little short gray DNA fragments, and this is the long read from the same subject. If you look at the short read, you don't really see much. If you look at the long read, here again, there's a big 300 base pair deletion that's pretty easy to pick up. You can see this drop in coverage also, which reflects that. And it turns out, I don't need to be sort of mysterious about who this might be. This is a super common deletion, actually. About 40% of people in our ADRC have this deletion. And what's interesting is this deletion is the red arrow here. So I pointed to these 4 variants that are always co-inherited that protect against frontotemporal dementia. And it turns out this deletion, this 300 base pair deletion, sits smack on this important part of the gene called the 3' untranslated region. And it's also in perfect linkage with these other 4 variants. So if you have one of these variants, you also have the deletion, and we can't disentangle those. That's making things a little problematic, but because this is a bigger change in the genome, and because it falls on a very important part of the gene that's important for determining where the protein gets made and how it gets made, we think this is the likely causal variant at this locus. We still need to prove that, and that's going to take a little more doing. But because you guys also generate blood that gives us proteomics, some of you generate CSF or provide CSF, we can look at proteins in the spinal fluid. Piecing these things together with these genetic changes will allow us to get to the mechanism. All right. So I hope I've managed to convince you that long-read sequencing is exciting. It's really turning up already new large genetic variants that we expect affect risk of Alzheimer's disease and related disorders, including Parkinson's and Lewy body. And I do think that we're leading the charge here. So we're going to start uploading these long-read whole genomes to an NIH website now. And these will be the first 400 or 500 sort of widely available long-read genomes. So I think that's a really big contribution, and wanted to thank you guys for that. And I think, this is just the beginning. I think we expect to find a lot more important findings related to these large structural variants from our long-read sequencing. So I think that ends the bulk of my talk. And this is really something that's moving quickly. So please come back next year. We'll have some more exciting structural variant news to tell you. Thanks. "11 minutes, I think. Sorry. Anyway, yeah. Wait for a microphone. They're gonna bring one around.
So I've been doing this part of this study for quite a while now, and I don't think that I've had any blood tests for a good 3 or 4 years. Should I encourage the people who I interact with to do that?
I guess Victor wants to field this one first, but I'll have a comment.
No, I wanted to protect you, but. So originally we're set up to try to bring people back every year and obtain blood every year. And that's sort of what we're circling back and trying to do. During the pandemic, no one was coming in. And there was a lot of reorganization in the research space that we had. And so we're a little bit slow in sort of coming back to that model, but certainly we want to receive blood is really valuable. Having it one time is valuable. Having it over a period of time, being able to monitor is even more valuable.
And despite that, my expectation is that your DNA was included here. The nice thing is the very first time you give blood, we can store the white blood cells, freeze them, and they can be, thawed, and we can do DNA on them ages later. I'm not afraid. We've got you. It's frozen. I'm sure we have it. It's the miracle of freezing DNA. Yeah.
Any other questions?
There's a question behind you, I guess. Here, I'll even just pass the mic here and we can share.
So if I understood this correctly, there's a gene, I forgot the names of it. It's guaranteed you'll get Alzheimer's from. Okay, that's not like Parkinson's at all then.
So I think you guys heard the question. There's a gene that if you have the mutation, you definitely get Alzheimer's disease. So mutations on 3 genes can give you that sort of early onset picture. There are genes like that in Parkinson's. So for example, GBA is a gene on which, right, so everybody's got the gene. And some variants on GBA increase risk for Parkinson's, but don't guarantee it. But there are a couple stronger ones that essentially guarantee it. But there are a lot of people walking around with GBA variants that will never get. So some of GBA is more like ApoE in Alzheimer's. It's a risk factor, but it doesn't mean you're definitely going to get it. But there are definitely a few mutations on GBA that are much, much stronger, more like the APP presenilin mutations. Yeah. One more question here in the back from Alicia. Yes.
Hi, I just thought I would share. I carry the PSEN1 mutation. My mother had Alzheimer's symptoms when she was 43 and she passed at 56. And if you want to know more about these genes, you can go to my website. It's called youngtimers.org, and it explains these 3 genes in detail.
Thank you, and thank you for coming.
And just to repeat her website, youngtimers.org. YOUNGTIMERS.org. Thank you for sharing that.
So if there are no more questions, I think we're at our break. Is that true, Nusha? Yes. All right. More caffeine. Thanks, you guys.
So this is a good time to get up and stretch your legs. Take a little break. I'll come around with my little chime a few minutes before we reconvene. And we'll go from there. Thank you. Let's do this. Why don't you all stand up a moment? I like to take advantage of having a captive audience. So I'm going to lead you all in a breathing and stretching exercise real quick, and then you're welcome to go get some more coffee. So here we go. You're all going to cross your arms down here. Inhaling up. Slowly exhaling out through the mouth. One more time. Bend the knees. Inhaling up. Exhaling slowly. Other arm in front. Inhaling up. Exhaling down. So 3 breaths in the morning, 3 breaths at lunchtime, 3 breaths in the afternoon, and you're good to go. All right, go enjoy your break. We'll be back in a little bit.
Second introduction. Hello, everyone. Let's come back. Let's sit down. I hope everyone is coffeeed up, and I'm really happy to have Dr. Weiss-Correa here will be speaking to us. Dr. Weiss-Correa was one of the founding members of this Alzheimer's Disease Research Center. He's played a really integral, high-level role, throughout the last, I guess, nearly a decade now. And he leads the biomarker core. He's also become involved in something referred to as the NIGHT initiative, looking at brain resilience, and that whole group is doing just really important additional work. The ADRC interface is there, but it's really a separate initiative. And he's here today to tell us about some of his work related to what can be discovered in the blood relating to aging, organ aging, and things of great interest to all of us. Thank you.
Thank you, Victor. Good morning, everyone. I want to start with really thanking all of you for participating in our Alzheimer's Research Center. You're really enabling the research that we're doing and moving the knowledge forward to hopefully put this disease in the past. So what we are interested in is understanding the relationship between the brain and the rest of the body. Neuroscientists have often focused just studying the brain, but we know that the brain is highly susceptible to what happens in the rest of the body. And I just list here a few examples. Let's say you have an infection, COVID or influenza, that affects your mood, it affects your brain, it affects your well-being, how you feel. Immune responses in general have an effect on the brain, but then also increasingly people realize what happens in the gut. That's particularly important in Parkinson's disease, for example. And then exercise is another example where we know that if you exercise, so you exert your muscles throughout the body, and that is beneficial for the brain. It also goes the other way. The brain regulates what happens in the rest of the body, and mood or stress are examples of regulators of the immune system, for example. So how you feel has a big effect on how the immune system works. And then, of course, we have general hormonal function. And really at the heart of all of this communication, how tissues in the body talk to each other and how they talk to the brain is in a significant part mediated by the blood. So the blood is this tissue that is at the center of all the communication. If you think about it, blood vessels go into every organ in your body. And so they have the ability to basically collect information. So what is in a drop of blood? if you look at the blood, just initially at a high level, there is a liquid in there. We call this plasma. This is sort of a yellowish fluid. And there's lots of cells in there, immune cells, red blood cells. But then when you look what is in that liquid, in that plasma, there's thousands of different molecules. There's fat molecules, there's proteins, there's sugars, all kinds of different things. Even DNA that Mike talked earlier about can be found in the blood that is floating around. We're trying to understand what that's doing. And traditionally, if you go to a doctor, they take your blood, and I really appreciate that you all volunteer to give blood because it allows us to do the studies that I'm going to talk about a bit more. But when you give this blood, then we have what we call standard clinical chemistry that informs the physician about basic functions of your organs. But what we want to do is really expand that and take advantage of these new technologies that allows us to now measure thousands of different proteins, for example, sequence the entire DNA or the coding of genes, the RNA, or proteins, the RNA. And this way get much more insight into what we currently do in basic clinical chemistry, so that we learn about the biology of the body, we learn about aging, but we learn also about function of different tissues. And we call this the molecularization of plasma. And this can, again, go across all kinds of different molecular entities. And again, what we want to do is to take blood from individuals at any age, look how the effect of genes, of age, of environment, of lifestyle, what you eat, how that influences the composition of the blood when we look at these thousands of different things. And again, we focus on proteins. So what we did in one of the studies that I'm showing you here, we obtained blood samples from individuals, roughly 4,000 healthy individuals from 20 to almost 100 years of age. And then we measured 3,000 different proteins. So these proteins might be involved in regulating immune function, hormonal functions, but they could also be structural proteins. Any type of function that happens in our body is basically performed by proteins. And we have about at least 20,000 different ones. And so here we measure 3,000, and we sort of look at all of these proteins across the 4,000 people, and we use colors to express the changes here. So blue means low level of a specific protein, and yellow would mean high levels of a protein. And so in this graph, what you see is you see these 4,000 people from left to right. And then for each person, we measured 3,000 proteins. So this, you can't see the individual dots, but these are actually 3,000 measurements for 4,000 people. And what you can immediately appreciate is there are dramatic changes as you age, and not just when you're old. When we typically say somebody is old, although that keeps shifting as we reach higher and higher life expectancy. But even when you're 20 or 30, there's a lot of changes that happen compared to when you're 15. We don't have this on here, but we can actually count how many proteins change at each age, from younger people to older people. And what we observed is these curious, what we call waves of aging. There's a dramatic change that happens at around age 35, then another wave around age 60, and then the largest wave at around age 80. And so these are dramatic changes that happen in our body that we can see when we measure these thousands of different proteins. So this is basically a reflection of aging, if you will. And it tells us that age is not a linear process, so it doesn't go completely up and up and up and up, but it goes in waves. There's something happening dramatically in these young people. So if you can tell your children or your grandchildren, you know, you're actually aging dramatically when you're 35 years of age. And maybe that actually predicts something for the future, how you're going to age in the future. And so one thing that people have started to do, if you look at this, you know, picture, this Roy Lichtenstein type painting, if you will, you can take any time point and you can pretty much say this is a unique signature, right? And so this is what we can do here. So if I take the person here in this red rectangle, I can pretty much say where that person belongs to, right? So it's almost like a signature of aging. If you give me your sample, I can tell you how old you are. And this is indeed what people can do. So we call this machine learning. It's not really artificial intelligence. It's relatively simple machine learning. But that allows us to collect information from lots of people and then compare them. So if you get a new sample, you can basically compare it to where does it fit in and say, okay, this person is 28 years of age. And so this information is not just telling us how old the person is, more or less, but it has biological information, right? Because there are all these changes happening with aging. So it tells us something about the aging process in itself. The level of each of these proteins has something to do with how people age, and potentially how your organs age and how your organs become susceptible to disease. And this is exactly what we do. So when we measure these 3,000 proteins, for each of them, we can ask, where in the body is it produced? Remember, we measure it in the blood, in this liquid, the plasma, but they're not coming just from blood cells. These molecules are collected from throughout the body. And in fact, so this is a slightly larger study where we measure now almost 5,000 proteins. We find 200 proteins in the blood that we can say with high certainty, they come from the brain. The brain is producing them and somehow they make it into the blood. So if we measure the levels of these proteins in the blood, we can say something about the brain. Isn't that amazing? So if that's true, then the blood sample that you give us will allow us to tell us something about how your brain functions, how well it functions, and how it might function in the future potentially. And that's exactly what we do. So we call these aging models, or clocks, how they have often been called. You may have heard about so-called epigenetic clocks that read changes in the DNA. But these are much more specific because they measure proteins from different organs. And so you basically then have the actual age of a person, and then we use this model, this machine learning model, to estimate the age of the sample. And sometimes we hit it right away. A person has the age exactly that that they are, so we predict it exactly. And sometimes there's a difference, so we call this a gap. And what's really interesting is that gap, that prediction, whether your brain, for example, is younger or older, is a good predictor of cognitive function and cognitive decline in the future, as you will see. So what we can then do is with these proteins in the blood that come from all kinds of different tissues, we can estimate the age of each of your tissue. So we can tell you, you have a kidney that is your exact age, or it's slightly older or slightly younger. We can do this for the liver, for the heart, for every tissue. And here I show you an example for the heart. So again, this is the actual age of a person, and then we predict the age of their heart. And you can see some people are exactly on this line, and others are a bit older or younger. We know which proteins are. It only uses about 8 proteins here. And then we can predict, if we get the sample at time point 0, here we have 15-year follow-up, we can look at people who have a young at heart. They have a very low risk of getting a heart attack or getting other heart disease, atrial fibrillation, for example. Whereas people who have an older heart, predicted to have an old heart, have an almost 3-fold increased risk to develop heart disease. So we look into future here. And we can do this for many different organs. And when we look at this, so here we predict all the different organ ages. And we have 3 individuals. They're all predicted. If you just look at their overall age, they're all predicted to have the exact age. But if you look at the individual organs, this person has all of their organs are a little bit younger. Here it's sort of some are younger, some are older. And then this person has most of their organs a little bit older. But what's really interesting, if we look at those who have very old tissue, and we call something that has 2 standard deviations above the average. So we call these extreme organ agers. We find here, for example, a person has an old kidney, much older than it should be. A person has a much older heart, and this person has multiple tissues, much older. And here we just project this on body map. So this person has most of their other tissues have the exact age that they are. So if this person was 75, most of the organ ages would be predicted to 75, except the kidney. And we find about 1.5% of healthy people in the population above 50 belong into kidney agers. About 1.8% are heart agers in our studies, and then 1% are these multi-agers. And these are 6 times more likely to die in the next 10 years. So there's good predictions for all of these models. And lastly, for the brain, we can do the same thing. So again, we have the age of a person, we predict the age of their brain. And here we have 5-year follow-up. And this is actually data from the Alzheimer's Research Center here. So we have blood from many of you when you were when you initially started to join our ADRC. And then we have follow-up. And so we find people with younger brains, people predicted with older brains. And again, those with the older brains are at much higher risk to show cognitive decline over 5 years than those with young brains. And what we also find, and this supports what other researchers have discovered, is that people with Alzheimer's disease have older brains when we use these metrics. So consistent with this idea that your body, not every organ ages at the same rate, and those organs that age faster will probably be susceptible to disease. And so hopefully we can see this much earlier than before you have disease, to start to do interventions and hopefully develop also drugs that target these specific changes. With that, I would like to thank all my colleagues here at Stanford, collaborators elsewhere, and then my lab. Thank you. Yep. Oh.
Tony, is there any possibility that this test is presently available?
Not yet. I mean, it's still in the research state, but we're trying to see, you know, right now, this is based on about 6,500 people. So we want to have 50,000 or 100,000 people to have really solid data. And then I think it also needs to be interpreted. It doesn't help you if you just know my kidney is older. You want the physician to tell you what to do about it. Yeah. But great question.
In your chart of the age peaks, particularly with the talk coming up this afternoon, are there gender differences, or are there sort of population differences, or are humans generally falling into those patterns?
That's a great question. So we see this in male and female, and it has recently been confirmed with a different technology that they see the same thing. But there are different proteins in these peaks between men and women. Unfortunately, we don't have enough data to look across different populations, but this is definitely will be possible soon. So this idea of measuring thousands of proteins has really caught on, and there's multiple studies now that were just published in this last two or 3 weeks with 50,000 people from the UK Biobank. And so we want to get our hands on these data, and we'll have hopefully better answers for that question.
Yeah, on the slide that had the brain age prediction, cognitive decline, and tissue age, it looked like the brain was more widely dispersed or fuzzy. Is that my perception, or is that a reality?
It's probably a bit too early to say, but yes, we have definitely a lot of differences. And in part, it could be the models, but maybe there are also many more ways how the brain can get impaired than, let's say, the heart, for example. Yeah, thank you.
So my question is also about the slide with the peaks. And I'm particularly interested in the green line, the one around age 80. Are there behavioral and experiential changes that you can associate to those blood markers? Top three, let's say.
At this point, for this particular study, unfortunately, we didn't have enough information on function of these individuals. But again, that will be possible in the near future, I think, where this UK Biobank is a large population-based studies with very deep phenotyping, we call this, where we have a lot of information on people. So hopefully we will start to be able to really tie the changes to function. Yeah.
I was just wondering if the aging of one organ, let's say you have a kidney that has an age that's quite high, will the aging of the kidney affect, let's say, the aging of the heart or the aging of the brain?
That's a fantastic question. We believe so. So we've shown earlier that if you just take the plasma of an old mouse, for example, and you put it in a young mouse, you can make that young mouse older. And vice versa, maybe more interestingly, if you take the young blood, even from young humans, and you put it in an old mouse, you can regenerate tissues and you can make them function better. But it's also true, people have shown, if you induce aging specifically in one organ, you produce inflammatory factor, but also other proteins that we know less about what they do, that then almost infect or propagate aging throughout the body. So it's all connected. Yeah. Great. Thank you.
So while we're getting the slides set up, let me briefly introduce our next speaker, who is Dr. Birgit Schule. She is a physician scientist coming from the Parkinson's disease world, but we sort of dragged her a little bit over into the Alzheimer's disease world as well. She is the person who spearheaded a lot of the stem cell activities that are going on within our Healthy Brain Aging Study Alzheimer's Disease Research Center. And so those of you who have been able to provide skin, have donated skin, and for those of you who don't know, it's a tiny little, smaller than a pencil eraser taken from the thigh usually, but those skin cells can be used for a lot of other things that are surprisingly brain related, and that I think is what you're going to hear more about.
Thanks, Victor, for the nice introduction. Can you all hear me? Okay, good. So my talk is entitled Why We Ask for Skin Samples, and my informal title is Why Do You Want Skin in the Game? So most of you have provided a skin sample, and you might have thought, what happens to my skin sample when it's in the lab? And I'll show you a little bit how that looks like, and you're also welcome to see the lab and actually kind of experience how it really looks. Okay, so when the skin sample has been taken, it comes to the lab and we are processing it. And let me see, I can use a laser pointer. So it's a teeny tiny piece of skin, and in the lab under the microscope, we cut it into much smaller pieces using these scalpels. And then we put these little pieces into tissue culture dishes. So I also call this Parkinson's disease or Alzheimer's disease in a dish. And when I say dish, these are these plastic wells where we put the cells in. And you see this liquid, this orange liquid. One of my sons was called this Orangina. So basically that's what it is. So it's a liquid which has sugar and other nutrients to how these cells grow. And then you might wonder, where do those cells live? And the home of these skins is in these incubators. These are at 37 degrees Celsius, which is body temperature, and that's where the cells grow. When we don't use the cells and we say we bank the cells, they're in containers that have liquid nitrogen, which is a very cold liquid, minus 190 degrees Celsius or 300 or so, minus 300 Fahrenheit. And so in these containers, these cells hibernate. They're in special media and culture media, and then we can store them for multiple years. So when you have given a skin biopsy like 10 years ago, we still have it. And these cells live in these containers with this liquid nitrogen. So they're not dead. And then you might wonder how they look under the microscope. And these are skin cells in a dish. What you see here, this dark part, is a piece of skin. And here you see these little spindle-like structures. Those are the skin cells that we're interested in studying. And then I might also say, your skin cells grow. And then we bank and share and make scientific discoveries. And how that looks under the microscope is like this. So for example, in one biopsy at day 8 after biopsy, you have these little structures, these individual structures, each of them are one cell. And then you can follow them over days under the microscope. And you look at them every other day. And as you can see, they multiply. So they're more at day 10 and at day 8. And at day 12, we would call them confluent. So they're so dense in the tissue culture dish that they are confluent. So there are no more cells that are fitting in there. And it's with your skin. So at that time, they don't grow anymore. They don't divide anymore. So then they're confluent. So growing means dividing, multiplying, and then we store them and they can hibernate. So, but now you might think, oh, these are skin cells, but we want to study the brain. So we want to study the neurons in the brain. So how do we model that with these skin cells? Oops. And for that, I need to go back and explain a little bit about development. And what you see here on the x-axis, you see different cell types. And here, the time is modeled. And we thought when we would start with the stem cells in our body, this development was only unidirectional. You would start with a pluripotent embryonic stem cell that would then differentiate into precursor cells or multipotent cells, and then ultimately mature into the final cell type, like the brain or the liver, the heart, the kidney. And so that was really kind of a dogma in biology. However, for the longest time, hundreds and thousands of years, people were wondering, is there a fountain of youth? So can we return that clock, yeah? So this is a very nice picture of Lucas Cranach about 500 years ago. Old ladies are coming here on cards and carried. Then they jump into the fountain of youth and they come out as young, beautiful women again. There are no men on this picture. I don't know why. Men don't age, probably. And if you think about it scientifically and use scientific terms, and what was discovered about 15 years ago is something that we call reprogramming of mature cells into induced pluripotent stem cells. That's basically the fountain of youth, all these complicated terms in kind of scientific terms. So now, going back to this model, we know now that cell fate is flexible and reversible. So we found the fountain of youth. And it's very interesting. The fountain of youth, you only need 4 different factors in this water, basically, to turn the clock backwards. And as I showed you, the cell fate is flexible, so we can reprogram the cells back into these stem cell-like cells in a dish. And that's what we're doing with your skin cells. So how do these cells look under the microscope? The skin cells I've shown you before, they form these wavy-like structures, and they're small and spindle-like. But when they become stem cells, they grow into these stem cell colonies. And each colony consists of thousands of cells. So here I just kind of try to delineate one single cell. And for these stem cell colonies, this little red dot represents one cell. I know this is sometimes really difficult for non-scientists to envision these terms, so I wanted to illustrate that. So then what can these stem cells or induced pluripotent stem cells be used for that originated from your skin cells? So this is about modeling neurodegeneration in a plastic dish, in a Petri dish. And so here, the model again. So we're starting with these induced pluripotent stem cells, and we want to get them into neurons. So we call this in vitro differentiation, making neurons in a dish. And so now we follow the timeline in vitro in the laboratory, turn induced pluripotent stem cells into specific cell types, for example, neurons, but also Mike earlier talked about microglia. It's another important cell type in the brain. And what we need to do is we have to mimic the human development in the laboratory, in the dish, to be able to then differentiate into neuronal and non-neuronal cell types. And this is very complicated, and I didn't want to go into that much detail because you can spend your whole scientific career on that. So I just call this, we use scientific pixie dust basically to do that. These are very, very complicated protocols we have to use. So now we have neurons, and you might wonder how neurons look under the microscope. And this is the stem cell colony you already know. Then we differentiate over several weeks or even months by really attending these cells every other day. We're changing media, we're changing the factors that we are putting into this media, and we're basically kind of recreating the development of a human being in a dish. So that can take quite some time. And then in terms of neurons, you see these, every little dot is a cell, and here I just kind of drew or outlined how a neuron looks like. Okay. And then here I wanted to put everything together, the strategy, what we're doing. So we're starting here with a skin biopsy. We turn these cells into iPSCs that I just showed you using the fountain of youth. We have them here, and then we differentiate the cells into different brain cells using the scientific pixie dust. And it has become quite sophisticated. So we can make neurons, we can make these other non-neuronal cell types. They're called oligodendrocytes, astrocytes, microglia. We can also look at some earlier progenitor cells that might be also interesting for some of the young onset forms of Parkinson's disease and Alzheimer's disease. And when we have all these cell types in a dish, we can also put them together as 3D cultures, which we think is a much more physiological. And then a lot of effort has been ongoing in terms of the models that we can do. So we want to look at what are the differences between individuals, cells from individuals with Alzheimer's disease, Lewy body dementia, Parkinson's disease, and controls. So that's why we need controls, very, very important. And we do mechanistic studies, and we also can do drug discovery with it. And so in the end, we are hoping to have a better understanding of neurodegeneration, but also being able to use these cultures for drug discovery and developing novel therapies. So within the ADRC and the healthy aging study at the ADRC, we have this portfolio. So we are starting with patient samples. Then we go through this stem cell generation. We have to do a lot of QC efforts to make sure these stem cells are good. And then we do this in vitro modeling that I was just explaining, using a lot of microscopy techniques. But we are also doing like the biomarker comparison. So we can use information that we have through the biomarker core and Tony's group looking in plasma, and then comparing that to what we see in vitro in the dish. And so we use the supernatant of this medium as a comparison, and we can also see the biomarkers that we see in plasma or in the CSF also in these cell culture models. So the advantages over other models for induced pluripotent stem cells is that we have a human genetic background. They're coming from patients with the disease. Some of them have genetic mutations or risk factors like ApoE. In the dish, we can also model some environmental factors that we believe have some increased risk for disease. And then genetically, we can also introduce and manipulate these cultures. So we can introduce additional mutations, or we can also correct those mutations and see if there's causality. And then looking into the future, the next frontier is really modeling the brain on a chip. So using these biological models and then combine them with engineering of chips to have additional readouts so that we understand better the physiological changes. We can also model the blood-brain barrier, combine the brain with different other organs on the chip. And then hopefully this can also in the future replace some animal models that we need to use at this point. Okay, that's the end of my talk. I want to also thank the ADRC iPSC team that's working with me. If you have questions, there's my email. I also have a Twitter X handle, and I thank you for your attention. Thank you.
So by the time we have this conference next time, should we expect all of you to look younger?
I'm just envisioning kind of my hot tub and then sprinkling and stuff. Maybe. Hopefully. Thank you for that question.
Does it matter what type of skin cell you start out with?
That's a good question. Does it matter what type of skin cell? It doesn't matter. We can also do this or apply the fountain of youth also on other cells. We can also do that with the white blood cells in blood. So we're doing it on both. So basically what you can do is you can take any cell of the human body and reprogram that into induced pluripotent stem cells. It doesn't matter. But one thing that does matter is when you take skin, skin is exposed to sunlight, and that is inducing DNA damage on the genome. So that can show us some changes that are not due to the underlying disease, but it's introduced to sunlight. So in that sense, it does matter. So I like to take the biopsies from mostly unexposed areas. So like under the arm, like here, it's mostly unexposed to direct sunlight.
So I'm understanding there's some gut biome research that's going on. And when you said environmental, I wondered if you could give us some examples of that from the environment.
Yeah, for Lewy body disorders, for example, pesticides increase risk. That's something that we can model. So you can expose these cultures to certain pesticides, solvents, another one. So that's relatively easy to do because you can just kind of add it to the culture.
So this is kind of a mechanical question. When you have your individual cells that you're starting with in your petri dish, what do you do to make them turn into cell kind A or cell kind B or cell kind C or cell kind D? Because I gather you can turn this A cell into a multiple of different kinds of cells.
That's true. So depending where you start. So with the skin cells, we first have to make them pluripotent so that they have the ability to then differentiate into the cell type of interest that you want to study. And then I didn't go into that much detail, so I call it just scientific pixie dust. So we know specific formulas that turn a pluripotent stem cell into a neuron or into a heart cell or a kidney cell. So they're different recipes, basically. Yeah. So to turn a skin cell into a pluripotent stem cell, you need 4 ingredients. If you turn a pluripotent stem cell into a neuron, you probably need 15 different ingredients, but also not all at the same time. So you have to introduce them at different time points during that differentiation process. That's very complicated. Almost every other week, you see in the literature new protocols published. So all these recipes get refined kind of on an ongoing basis. Okay, thank you. I think we have.
So thanks so much. I would point out that this kind of research is not being done at all the ADRCs. It's being done at a couple of others, but importantly, the resources that are being developed here, they're being developed by Dr. Schulin, and the people she's working with are being shared on a national basis through a coordinating center at Indiana University. And so this helps science not only here at Stanford, but also at other universities and other places throughout the country. And so the skin that you give here, your skin in the game is in the game nationwide and maybe globally, I don't know. So I think there is a lunch coming up and Nusha wants to tell us about how
No, not
what
yet.
we're doing.
I would like to ask all the speakers to please come to the front. We now want to open this up to the Q&A session. Our wonderful helpers over there have collated the index cards you've all given us. So if you want to, Leah and others, if you want to come up and you'll ask the questions and the speaker and then direct it to the speakers, and then we can have people answer. So this will probably become a shared mic.
So I guess we're not off the hook yet.
I can maybe say why there are no men on that fountain of youth. Do you know? It's because they thought that the men get rejuvenated by the young women. That is the official interpretation.
Now you're opening
It's a different time, yeah.
500 years.
Okay, that's right. Good. Hello. Okay. First, we have a question for Dr. Henderson. So regarding the slide on brain shrinkage from the anti-amyloid slide, I don't know if we can pull it up or not.
No, it wasn't really a slide on it, but it's a mention of it, so we don't need to pull it up.
Okay, so a participant was just wondering what exactly is what do the percentages represent? What do they mean? Is it brain shrinkage?
No, it's a good question. So I think all of us know, in the absence of some of the factors that Tony is developing that will make us all younger next year when we all come back, that there is on average some shrinkage of the brain with aging. This is part of the usual aging process. There's more shrinkage in pathological conditions like Alzheimer's disease. And so in these studies, it's not surprising that over time there's a little bit of brain shrinkage. It occurs in people who are actively treated with these new medications and people on placebo, but there was more shrinkage in the group that was actively treated. And I think the percentages that were up there for the different studies were like 25%, but that represents a 25% difference in the rate of shrinkage rather than, no, your brain is not shrinking by 25% over an 18-month period of time. That would not be good. But it is shrinking a little bit more in the actively treated group compared to the other group. People have done the calculations, I haven't, and said that the amount of amyloid being pulled out of the brain doesn't account for this relatively small difference, although in terms of percentage, it's sort of one of my medium-sized check marks rather than a tiny check mark. So that's what the numbers were referring to. And thanks for that question to clarify that.
Okay, so the next question is for Dr. Gracious, and is early AD hard to detect?
Yeah, it's a really good question. So I think once people are symptomatic, whether they're in their 40s or in their 70s, it's a little easier to detect. I think early onset Alzheimer's I think a lot of those patients get sort of knocked around a little bit because people don't expect people in their 40s to develop memory trouble, right? And so they might be told that they're anxious or they're depressed or something, and they may see a psychiatrist first. That's a pretty common story. Once they get to us, even though they're rare, we're used to seeing patients with early onset Alzheimer's disease. And with these biomarkers now, the sort of diagnostics can get done at earlier and earlier, even preclinical stages. But for the typical clinical presentation, I think there's often a delay in early onset Alzheimer's, especially if there's not a well-characterized family history and people aren't out for this.
Okay, and can I use your microphone? Okay, and this next question is for Dr. Wise Corey. Have plasma donations of young people been studied?
Yeah, great question. So there's actually a lot of plasma being collected for clinical purposes. So there's companies who collect daily thousands of donations from volunteers, and they produce, for example, antibodies that protect people with immunodeficiencies. Albumin, if you have a lot of blood loss, you may get fractions of blood from these individuals. Increasingly, people are starting to analyze what is in the blood from young people compared to old people, as I showed you, and trying to see whether that tells you something about diseases and aging. But yes, these samples are being studied.
Great. Thank you. And this is another question for Dr. Henderson. What effect will artificial intelligence have on cures for neurodegenerative diseases?
So I'm artificially intelligent. So AI encompasses sort of a broad category of new approaches, and I think it's really powerful. It's extraordinarily exciting in a lot of ways, maybe a little bit scary in other ways. And I think the broad answer is these techniques are going to be applied to a lot of different scientific questions, including related to causes, treatments, new insights into neurodegenerative disorders. And I think ultimately the insights will be really powerful. I'm not sure how quickly they'll come. I think probably fairly quickly. One of the things to be cautious about is that we don't always know how these algorithms come up with a final solution. It's sort of based on data that goes in, but sort of what goes is between the input and the output is a little bit opaque. And so all of us need to be careful about what the interpretation is and what the insights are, and to validate those as they emerge in other ways. And Tony, maybe you have other comments or thoughts about this as well?
Yeah, I think one example is that artificial intelligence can learn about data without knowing what the data really are. And an example of this is, so I showed you we can measure thousands of proteins, right? If you use AI and you give the computer thousands or 100,000 different samples, it can basically recognize them and sort them the way we sorted them here. But moreover, it can learn what the relationship is between each protein to all the other ones. And that's basically biology. So in a way, the computer will be able to understand biology and life in general based on very rich data that we're starting to generate. And people have already made examples of that.
And I would put quotation marks around understand.
As good as we can.
Okay, this is another question for Dr. Gracious, and it's, is 23andMe a reliable source for detecting early onset AD?
I always feel like this is a plant each year. So I'm a huge 23andMe like anti-enthusiast. I would not get your genotyping done by 23andMe. You know, so first of all, no, they don't really look at some of these really damaging mutations for early onset Alzheimer's disease. The thing that they will offer is ApoE4 genotyping. I can tell you that we've had, you know, participants come through who found out their ApoE status from 23andMe. When we confirm it with our research genetics, we've never really seen any errors. So I don't think that it's incorrect. I just think it's a crummy service. It doesn't come with any counseling. Oftentimes, we have lots of people who discover that they're carrying 1 or 2 copies of ApoE4 accidentally through 23andMe because there's a 8-page consent form. You tick all the boxes, you don't read anything. And 6 weeks later, you find out that you have 2 copies of this incredibly powerful risk factor, right, out of the blue. So hate's a strong word, but I kind of hate 23andMe, and I wouldn't recommend using it for anything. If you want to, if you're interested in Ancestry, Ancestry.com is fantastic. Yeah, forget about 23andMe.
One thing I might add is that when you go into your doctor and you have cholesterol tested, for example, their drugs are really powerful in reducing cholesterol, and it's known that high cholesterol is a risk factor for stroke, for heart disease. So the good reasons for wanting to know what your cholesterol is and good ways to take care of it if there's an abnormality. We're not at that stage yet for knowing what to do with things like the E4 allele of apolipoprotein E. There are drugs that are being developed, but I think we still need to wait, and all of Dr. Gracious' caveats are really important to keep in mind.
Just one more little bash against 23andMe is that, so they've shared their data a fair bit in Parkinson's disease. I think Parkinson's compared to Alzheimer's is a little easier for people to self-report correctly that they have Parkinson's. And so 23andMe has contributed a lot of data. They're very uncollaborative, at least in our experience with looking at Alzheimer's data or just sharing their genetics. So it's not even great from like a citizen science standpoint.
And I think it's part of their business model. They're not, they're making money from running these tests, and that's fine. That's what they're set up to do. But they also use the information they collect in a lot of other ways, the way a lot of other businesses do. And this isn't inherently good or bad, but the reason why they hang on to a lot of the information they collect is part of their proprietary models and other things that they're interested in. Maybe, Mike, you know more about that than I do. Next question.
Thank you all. So this question goes out to everyone. How does prevention compare with current treatments for AD?
So, one of the big faults that's been levied against the anti-amyloid antibody trials is that we're not starting early enough. So we started in mild to moderate AD, Then we moved to, mild AD, and now we're in mild cognitive impairment and mild AD. And we still see these kind of very kind of underwhelming differences between groups. So increasingly now people are actually moving into preclinical Alzheimer's disease and prevention trials of a bunch of drugs, including the anti-amyloid antibody trials. The first big one to come out was published just in September in the New England Journal of Medicine, looking at one of these antibodies called solanezumab. That was negative, unfortunately. It was a 4-year study. These studies are really hard to do, right? People who are healthy don't decline as quickly as people that already have symptoms. And so these trials are very long, they're very expensive, they're hard to run. The first big one, at least in sort of late onset Alzheimer's disease, was just published and was completely negative. But there are others ongoing with, for example, lecanemab is being looked at in a prevention study. They've done this in late onset Alzheimer's and also in early onset with genetic mutations. Thus far, nothing's hit pay dirt, but I think it's an important part of what we want to look at.
Great, thank you for that. And going off of your answer, I was wondering, this is for all of the doctors as well, given the cost and outcome of the anti-amyloid drugs, do you believe the treatment is worthwhile?
Yeah, I mean, I'm among the bigger skeptics of these anti-amyloid antibodies, at least certainly in our, group and division. So Victor spoke a lot about this sort of, minimal clinical difference, that sort of important, so that at a year's time, if I saw somebody a year ago and I saw them today, you need to move by about 1 or 2 points on these scales that they use. That's in a year. These studies are 18 months long, so it's longer than a year. The biggest change we've seen is just over 0.5 point, right? Well under what we would expect would be noticeable. And I'm not even sure I believe those differences. So Victor talked about the functional unblinding. When people have brain swelling, the dosing's halted, they get more frequent MRIs. The physician knows, the patient knows, the care partner knows, right? The blind is broken. The CDR sum of the boxes is a very subjective scale. Everybody wants, their partner to get better. So if you think they're on the real juice, you might sort of subconsciously over-report the effects, right? That's the risk of unblinding. So I think these tiny effect sizes, when we correctly account for unblinding, are going to be tinier still and potentially even non-existent. I'm not going to be using any of these in my practice until I see better data. So no, I don't think it's worth the costs in time or money, but maybe somebody else has a different opinion. I know Victor doesn't really, but. Tony, what do you think about
I'm not
anti-amyloid
qualified. I'm not qualified to, judge from a clinical perspective. I think what I'm excited about is that some of the biomarkers are moving in the right direction. And maybe that's telling us that there is some biology that we're changing. And as we move in that direction, we may discover more powerful drugs that are based on the same idea, or with some of these diagnostic tools that we can catch people much earlier. We can also figure out what type of Alzheimer's disease a person might have. So I think most trials currently cannot distinguish what is really wrong in your brain, because we can only look at it structurally or based on, how you respond to these, as Mike said, not so good questionnaires. But we don't really know what's going on at the molecular level in the brain, and in which brain region you have most of the damage. Is it inflammatory? Is it more you have loss of neurons or the synapses? And I think as we develop better biomarkers that look at this at a molecular level, some of these drugs might actually work, or even other classes of drugs that we threw away because we can't really run good clinical trials might actually work. So I'm very hopeful that this revolution that we have with molecular biomarkers will give us new ways to maybe revisit some of these drugs.
Okay, for all speakers, will you comment on the Bredesen protocol for treating and preventing Alzheimer's?
No.
Who asked that? Who asked that question? Okay. So Dr. Bredesen is a well-known, well-respected neuroscientist who developed a protocol for, I forgot the name of his book, which I actually read, The End of Alzheimer's Disease. And the first part of the book I thought was really well done. I think the science is there. And then it becomes a lot more speculative. And as I recall, there were like 36 or 37 or 38 different things that people need to do, some of which are common sense, some of which have scientific validity, some of which are really not very well explained. And there's the warning, if you don't do all of these or all but one of these, then nothing will work. Some of the other things that he advocates, if I'm recalling correctly, involve proprietary testing. So I think there might be some potential conflict of interest in some of what's being recommended. The anecdotes that were presented in some of the publications really are not scientifically convincing, at least to me, and others may feel differently. So I think that's probably not the direction that most people would want to go in. Dr. Gracious.
Just save your money. I really don't think there's enough science behind it to merit spending your money and time on that book.
Okay, gotcha.
I read a borrowed copy. I didn't spend.
Okay, thank you all. And this is for all the speakers. What sort of support does the ADRC at Stanford provide for their participants?
So it depends on what's meant by support, and it's a good question and it's a fair question, because this center is set up primarily to advance the science, to make fundamental discoveries, and other things as well. And we're not primarily a treatment, a study. That's not the primary purpose of what we're doing. And so we're not saying that by doing this, we're going to cure you. But what we're saying is we're trying to find answers that will cure your children, prevent Alzheimer's disease from developing in your children or developing in you if we're sort of lucky and come to a really powerful solution within the next several years rather than a little bit further along the horizon. So, you certainly have our deepest appreciation knowing that we aren't able to make promises like, oh, if you take this drug, you'll be the first person cured of Alzheimer's disease, because that's not what we do. We try to provide information about the disease through conferences like this and through other means so that people know what's going on in the field. We've had this ongoing internal debate about what kinds of research information should we send back and provide back to our participants. And I think maybe we haven't done quite as well as we might. Sometimes interpreting memory tests, for example, done within a research setting, using research tests that aren't fully normed, it's sort of hard to know what the right interpretation might be and how to give this information back in a way that wouldn't cause more anxiety than reassurance. We do let people know if there are cognitive changes that they aren't aware of that maybe should be checked into more carefully. We've been working out a protocol so the results of testing like ApoE is available for people who want those results. We're able to provide that. We are able to provide copies of some of the research imaging that's done, MRI scans and PET scan results, if people want that. Some people do, some people really don't want to know that. And so we're trying to provide more back to our participants. We're welcome to input and suggestions on how to do that. We have members of the Community Advisory Board and our outreach group are trying to think of new ways and better ways of reaching out and providing resources and support and information to members of the community that we're recruiting from, including parts of the community that aren't well represented in research, and we're trying to recruit more from. So I hope that gives you at least a partial answer about where our thinking is and what we do and what we're trying to do.
Great, thank you. And we have one last question before we break for lunch. And this is from Dr. Henderson's slides as well. So for patients taking anti-amyloid drugs that experience the amyloid-related imaging abnormalities, what are some of the symptoms they experience?
So what is pointed out often, which is true, is that sometimes these effects that can be seen pretty readily on imaging, like the MRI brain scan, aren't noticed by participants. There might be some underlying harm, there may not be, but they aren't always noticeable. The edema can show up as confusion, as headaches, and in which case then the treating physician should then order an MRI scan to see what's going on. Is this swelling? Is this bleeding? Is it something unrelated? One of the difficulties is people with dementia sometimes become confused more often than other people, or everyone gets headaches. And so it means that a lot of things are going to be investigated that aren't really due to ARIA. But I would say a common effect of the edema, the swelling, would be headache and confusion is sort of a common manifestation. For the hemorrhages, if they're tiny, if they're microhemorrhages, probably there is a cumulative effect in the brain, but individually, they're not going to be noticed by participants. There can be larger hemorrhages, larger bleeding into the brain, which can be life-threatening. And in the donanemab trial, there were a couple of deaths that were attributed to the drug treatment. Fortunately, these are rare, but it's another reason why the onset of headache and confusion needs to be looked at carefully, because if it's more than a microhemorrhage, then it's something that needs to be taken care of pretty rapidly. None of these things are experienced by brains in the dish, by the way. So.
Can you all please join me in thanking all of our speakers one more time? Thank you all so much for your engagement. We greatly appreciate it. So before we break for lunch, there was one general question that I wanted to address, which is, are these slides going to be shared in any way? My hope and goal is once I get a confirmation from all the speakers that I'm able to post the slides on the website, I will be posting both the slides as well as this session is being recorded right now. And I need to do some editing, but I'll try and get it up before the end of the year. And I will send you all through the ADRC email that I normally send around the holidays. Hopefully by then I can let you know that the information is up on the website. Okay. So now it's time to replenish you and get those organs all revitalized again and get the blood pumping. So we'd like to invite you to join us for lunch outside. It is a buffet style. You're welcome to sit anywhere outside. There's plenty of seating, both in the sunshine and out of the sunshine. And then we will reconvene at 12:55. I will go around 5 minutes before that, and with my little chime to remind you it's time to reconvene. Enjoy, bon appetit, and we'll see you soon.
Yeah, I think that's part of what. No, that's why I think their focus has been more on TV. But just not a lot of and then they wrote You're still getting good. That's like a. And then blood going bad. All right. Yeah, I do. Yes, yes, that's what I'm doing. Well, that's another good question. Every model is wrong, so I usually have that slide up, but I didn't want to say so, but. It's never better than that's for sure. But in general, so one question I have to do, and I'm rephrasing this is an example of what great believer
Yeah, I know. At the NIH, as we pointed out, there's a question. One issue is cost. And it's cost and people that are doing. It is possible, and the NIH is pushing for the diverse IGSC very much so. In the end, it's a matter of how much time. It's all possible. It's all possible. It's like robots But how do you know with the skin cells? Because right now it's basically European dates. And is there something different? I had all these questions when you said something. I triggered all of that. Because I'm being pulled into this stem cell conversation. So can we truly mix everything? Can we just keep one diverse population going with blood cells and skin cells? And then there's another population that you have with this person because you don't know what the outcome is. Well, that's yeah. Not for a group.
So can I come to a partial answer first? That is, when you said that race is a construct, I think what's behind that is if you look at the difference is like tiny. So people, making their points based on the difference is so tiny. So a lot of the fundamental biology is probably the same. The other thing, there are a lot of things there are a lot of variation there. More restricted populations, some of the overwork. So we're missing that. Even in the functional and we're treated differently, it's more comorbidity, more illnesses. The way common genetic variation might be manifest in one setting where people aren't affected by pesticides or pollution.
As Parkinson's.
Then, they'll come out differently in another group it is, and so diversity is important. Even if it's not.
There's an article yesterday said that they're looking at pollution for Parkinson's. So okay, so the same type of thing, because I post every day, I watch it, all changes, so I want to be able to ensure that we're collective and wherever we go. We don't know what Parkinson's is and how it affects something like stem cells, because a lot of people want to go ahead and get that to grow those stuff, but we're losing on a regular basis.
I'm surprised that I didn't have that question.
I wasn't in for the whole thing. I
For
came from
replacement therapies.
Yeah, because that's big.
But I kept saying fish, so maybe that's.
Yeah. But if I had been here, I would have had something else going on. But I'm very pleased to meet. I'm sure we'll have our paths cross and all that.
Thank you so much. But to wrap something up.
Pleasure talking to you. Yeah.
Much more complicated slides. Came down to what is it?
My concern about clients. It's a drug study that's been showing actually. So explain what's going on. And I'm sure what it is. Studying all this. Explain this. Particularly the follow-up question where it might take 4 factors to send something back to the stem cell status, but it's a lot more than pathway differentiation. So say you do get Parkinson's disease here. Yeah. And it's got it right here. Well, I meant in terms of what people are doing. Like you may have been in my vaccine.
Sure, okay. It's all their weakness. And
It is well, I had somebody who hired as a consultant over South. The chairman of the board, Mr. Kennedy, was a consultant. He's looking at the vaccine. So there's a plumbing contract.
so what do you?
They got a plumbing contract. With the IRS obviously the increase of $1 million a year by default overcompensated. I like that. I'm absolutely serious. I think. No, if you're working for charity, that's, I was going to give him a chunk of money. And then I started researching it and said, wait a minute, they took like $1 million a year. Well, they since gave themselves raises of $340,000. Another 1.3 million. It's two people. Seriously. It's hard for nonprofits. Paying large salaries doesn't make sense. Go from being a nonprofit.
Well, I lived through high salaries at the Parkinson's Institute for the CEOs.
Was that part of the problem there?
That was a major problem.
But, you know.
Can I stop recording during lunch?
Yeah, feel free to pause it.
That was.
I probably should have done that.
Small.
Yeah, the two.
Having that knowledge is right.
Oh, okay. Yeah, no problem.
Oh, yeah.
I'm watching this time. Where were you before you CSF? Which one?
I was residency at Western University in Ontario.
Okay.
Yeah.
So I'm not sure from Nusha's signaling of me, whether we're supposed to get started or whether she needs to ring her chimes a little bit more. But I think we're near the time where we should get started. So let's come in, have a seat, and in 1 minute, I'll introduce our next speaker. Oh, the room is quiet enough. I think I'll make it 30 seconds instead of 1 minute. So thanks, everyone. Thank you, Nusha and others, for this wonderful lunch that we had. It's more than I was expecting. And my guess is there will be leftovers. And if there are, then I think everyone here should feel welcome to make do as best you can and take some of it home with you. So we're sort of continuing the theme to letting people know some of what goes on within our Alzheimer's Disease Research Center. And you heard this morning a lot about some of the marvelous things that can be measured in blood and plasma. But there's another fluid, and that's spinal fluid, that's more directly in contact with brain. And there are a lot of important studies, a lot of important measurements that can only be done uniquely through the spinal fluid. That's obtained through a technique called a lumbar puncture. It's really not very scary. And we have Dr. Ryan Taylor here to demystify, destigmatize some of what goes on in terms of a lumbar puncture. He is a faculty member here in the Department of Neurology. He came to us from UCSF, and before that from Ontario, which is up north. He's really been an important addition to our Alzheimer's Disease Research Center. And so, Dr. Taylor, thank you. Demystify what's going on here.
Thanks, everyone. It's a real pleasure to be able to speak to you all at once about this topic. It's something I'm, passionate about, and spend a lot of time in clinic talking to my own patients about. So, now that everyone's had some lunch and is probably a little bit sleepy from that, we're going to dive right into a topic that's hopefully going to wake you all up a little bit. So I'm not going to go too heavy on science and physiology around this, but I do just want to give some very basic background information. So cerebral spinal fluid is a fluid that lives inside the skull, inside the meninges, and bathes the entire brain and spinal cord. And so what you're looking at here is a image of the brain kind of from a side profile, and then the spinal cord down below it. You have about 150 milliliters of crystal clear fluid going all around the brain, and it doesn't just sit there. You're constantly making more of it, about 20 mils an hour. So it's constantly cycling through the brain, as the arrows and color changes kind of indicate there on this graph here for you. Because it bathes the entire brain and spinal cord, like Victor just mentioned, it contains a lot of helpful information about the health of the central nervous system. And LP is really one of those indispensable tests that every neurologist uses on a very, very regular basis. I would say that there are three in neurology. It's MRI, LP, and EEG. Those are really the indispensable tests that we have at our disposal in clinic. And it would be hard, I think, to practice 21st century medicine in a neurology clinic without this test available to us. We use it to diagnose serious illnesses like multiple sclerosis, encephalitis, meningitis, and now, more recently, even Alzheimer's disease and Parkinson's disease. Oftentimes, the diagnoses that we make by doing these tests on spinal fluid enable us to provide life-saving or life-altering treatments. And so it really, really is a critically important test. As I mentioned already, we do use this test to get more information about specific diseases like Alzheimer's disease, Lewy body disease, or Parkinson's disease. But those tests all came from research that was done in a lab to learn more about these illnesses before they could be used in clinical practice. And so that whole pathway is what you all make possible through your involvement in the ADRC, to learn more about spinal fluid, how it changes in disease, and how we can use that for earlier detection and develop treatments and monitor their success. But spinal fluid tells us a whole lot about the health of the nervous system and how that changes in various settings, including things like pregnancy, normal aging, and even systemic health issues like cardiovascular risk factors. And so all of that information is critically important. It's all stuff that we can detect through kind of research investigations into cerebral spinal fluid. So that's it. I'll leave the science there. I want to move on to talking about what a spinal tap or lumbar puncture is not. Okay. I wish this movie had never been made, not because it's a bad movie, although no comment, but because I think it is part of the zeitgeist and the stigma that is around this procedure. I personally wouldn't want to get a surgical procedure that shares its name with a fictional hair metal band. But lumbar puncture is not, it's really boring. I hope that's one thing that you guys come away with today. It's really a benign procedure. It's quite quick. There's not a whole lot to it. And I would say the number one feedback I get from most of my patients is, wow, that is not what I was expecting. It was much easier than I was expecting. I didn't experience any pain. And so we're going to hear a little bit more, some testimonial about that in a minute. But I think I'm qualified to speak on this topic for two reasons. One is that I'm a neurologist, and in my 10-year career in medicine so far, having spent most of that time in neurology, I've ordered and performed probably hundreds of these tests. But also in my younger days, I had a lumbar puncture done. I had a knee injury, and being a very eager pre-medical student right before the procedure, insisted that we not do general anesthesia, but do spinal anesthesia instead so that I could watch the procedure. And after the anesthesiologist let out an exacerbated sigh, he prepared me for spinal anesthesia, did an LP, and I just remember thinking at the time, wow, I can't believe how quick that was. My legs started to go numb before I knew the procedure had even started. So the lumbar punctures we do in clinic in the ADRC are a little bit different than spinal anesthesia, which some of you may have had experience with, and certainly some of you have experience with spinal tap, but the main difference is that we're removing fluid. So to run through the procedure really quickly, the neurologist performing your spinal tap will do it actually just in a regular clinic room. So we don't go to an operating room or a surgical suite or anything like that. We clean the skin really well ahead of time as a precautionary measure mostly. And then we numb the skin and the tissue in the low back. And after that, we really shouldn't feel any pain or discomfort at all. And I will say that the needle we use for the numbing part of this is quite small. Most people don't even feel that. Some people do feel a bit of pressure, but it should not be painful. And by the time we're collecting the fluid, most patients, as I mentioned, tell me that they weren't even aware that the procedure had started. The collection usually goes pretty quickly, and the whole procedure is usually wrapped up within 30 minutes. After the procedure, we usually ask people to just hang around the room for another 30 minutes to an hour. And the main reason we do that is just to give you enough time to replenish that fluid we took off. So I mentioned we reproduce about 20 milliliters of this fluid an hour. Usually in the ADRC, we're taking no more than that, usually a little bit less. And so usually within an hour, you've replenished all that fluid. And then after that, you're up and home, and usually there's no issues. I will say that the commonest side effect that only happens in about a third of patients is a headache. And in the overwhelming majority of patients, that headache is gone within the day. In extremely rare instances, it might last more than a couple days. The headache's usually pretty mild. It can be managed just with oral medication like Tylenol or Advil, and laying flat is usually enough to control or get rid of the headache. The reason being that the reason you might get a headache is because the pressure inside your head's a little bit lower than when we started. And so lying flat will equalize the pressure inside your head until you have enough time to reproduce the fluid. So that's a rundown of the procedure. I wanted to make sure I left lots of time for questions and some testimonial that we have here in a minute. But before I introduce our next speaker, did anyone have any questions about the procedure?
I do. So why is it impossible to paralyze someone? Because sometimes people are concerned.
Great question.
No, I don't want to be paralyzed.
Yeah, great question, Victor. Thanks for mentioning that. So if I go back to this slide, yeah, so the question was, why is this such a safe procedure, and why is there no risk of damage to the brain or spinal cord? And by virtue of that, no risk of someone becoming paralyzed from this procedure. On this image, you may not be able to see it super well, but the spinal cord is this black string in here, and it actually ends right here. And down here is a hollow fluid-filled space. There are some nerves kind of floating around in there, but this is where the spinal cord ends. We're usually doing the procedure in this area. Well, not usually, always. And so we're actually, very, very far distance from the brain and spinal cord itself. And so there is no risk of injury or damage to the brain or spinal cord. And so that's part of the reason that this is such a safe procedure to get done.
Thank you. Okay. Hi, as a part of this study, I did have what turned out to be an unsuccessful spinal tap. Should I get in touch with somebody and try again?
So I'll answer that, and then maybe Victor, if you want to chime in, but yeah, we're always happy to retry a spinal tap at a later date. I think usually the participation for most of you will be kind of an annual happening, and certainly at each visit, we would offer an opportunity to repeat that procedure.
Well, I wish it were that easy. Okay. One of the things that the pandemic did was that it limited in-person visits for here, but research across the campus and other universities, which meant that a lot of the in-person annual follow-ups that we wanted to occur didn't occur. And even before that, there was sort of a space crunch, and we in the clinic are sort of working that out. And so every year, we'd love to do it. And we're certainly trying to do other things every year, but for an LP, no. But the broader answer is yes. I think sometimes as people get older, there's a little bit of arthritis, and sometimes it's a little bit harder to direct the needle. Not everyone is experienced as Dr. Taylor, and sometimes because this is a research gift that you're giving us and not sort of a clinical test where it's really important to get the results, that some of our neurologists, if they are unsuccessful the first time through, will say, this is it, thank you, I'm not going to go a little bit further. But I would think, yes, it would be very feasible to do it again, and thank you for offering and considering a second attempt at that.
I came into the study a little more than a year ago, and I came in once and took a blood sample, and I was supposed to do a lumbar puncture, and then they decided that because of some factor in my blood, it wasn't quite as safe as it should be. And since then, I thought it's probably because of certain medicine I was taking then, I'm still taking now. I reduced the amount of that medicine, fortunately. I discussed what happened with my general doctor. She said, yeah, I'm not really worried about it. You're just kind of borderline. It's not like you're clearly unable, but they were being very safe. Should I contact that department and say, hey, do you want to try again? As I've heard you talk about how helpful this is, I feel kind of bad that they haven't been able to do it for me.
Yeah, so there are a couple of reasons that someone might not be able to get a spinal tap. One thing we do before every lumbar puncture is check to make sure that your blood is clotting properly. I think that's what you're probably referring to. Yeah. The reason we want to make sure your blood is clotting properly is so that you don't form a bruise when the procedure is done that could potentially push on some of the nerve roots there and cause some weakness or numbness in the legs that might need to be addressed through a second procedure. So we sometimes in the clinic will be a little bit less strict with that if there's a really strong medical indication to get a spinal tap done. But in the research setting, we usually want to be very strict because, as Victor said, this is not a clinically necessary test, and you guys are kind of donating your time and spinal fluid to us.
So I just let it rest. I'm taking less of that medicine now.
Well, I think, it certainly is something that could always be looked at again when you come back. And they could always just repeat that blood test. And if it's above the levels that we use as strict cutoff, then I think it would be reasonable to maybe consider.
And I think the caveat would be if it's one of these direct anticoagulants that even regardless of blood tests, probably
Right.
would be in the side of safety and not do that.
Yeah, there are some drugs that thin the blood that we can't measure really well how much they're working. Some of the newer drugs especially, and so when someone's on those, we can't use the blood test to say whether or not it's safe to get the procedure done.
Sorry, before we go to the next question, I just wanted to address for those who have questions or would like to come in and do a lumbar puncture. I did just check with our nurse coordinator, Christina Weiss-Corey. If you email the ADRC Stanford at stanford.edu email with that specific question or inquiry, I'll be happy to pass that along to her and then she can work with you directly. Okay, thank you.
Yeah, sorry, a very quick question that supposedly would that the lumbar puncture result would be clearly indicating something happening or may not be. Because my wife has dementia called cardactyl for about, this is her 8th year. At the very beginning, she did the lumbar puncture, but couldn't find anything. And suddenly, we really appreciate Dr. Gracious help to identify her have this genetic dementia.
So CADASIL in particular is an illness that usually cannot be diagnosed by spinal fluid alone. Usually requires a genetic test to confirm, as well as the constellation of clinical symptoms and the imaging findings. So in that particular case, spinal tap would be unlikely to yield a definitive conclusion, but would still be indispensable to rule out other possible mimics.
Hi, I have two questions. One is almost back to the woman who first said she had an unsuccessful spinal tap. What does the unsuccessful spinal tap look like? I mean,
Yeah, that's a great question. And I would say it differs a little bit in clinic than it would in research. So in clinic, for a medically necessary condition, especially if it's a very serious or life-threatening condition that we really need that spinal fluid to diagnose so that we can treat effectively, you may make multiple passes. So if the first pass of the needle is unsuccessful, meaning that you hit the path of the needle is obstructed by bone or connective tissue, degenerative changes, you'll try and make another pass. In the research setting, we're not as persistent. And so if we're not successful in the first or second pass, usually at that point we'll say, you know what, this is no medically necessary procedure. And so we're going to call this an unsuccessful attempt.
And I have a similar thing for blood draws. I have the 3 strikes and you're out.
That's very fair.
The other question I had is, I'm imagining the spinal cord coming out with all these nerves coming out the sides.
Mm-hmm.
Where does the sac that the spinal cord is in end, and how does that kind of work? Do you know what I'm saying? You've got all these holes in the bag then all of a sudden, with the nerves coming out the sides of the spinal cord.
Right.
Yeah, if the spinal cord's in a bag, is the whole nervous system in a bag? Am I clear now?
Yeah, so the sac that the spinal cord sits in ends shortly after the nerves come off. So it follows those, and then through cellular adhesion and connective tissue will seal that sac. So it's not an open system. It's not leaking spinal fluid out from those holes where the nerves come off the spine. It's sealed. Yeah, exactly.
Would severe central canal narrowing or stenosis at L3, L4 impact this?
No. So degenerative changes in the spine, prior back surgeries are not a contraindication to spinal tap and oftentimes don't make it more difficult. I will say that certain back procedures might actually make it easier, especially if you've had a laminectomy. There's just less bone there in the back. But no, a history of back surgery or degenerative changes in the spine would not be a reason to not get this procedure done. Yeah, so taking up a lot of time here. I appreciate everyone's questions. I do want to introduce our next speaker who's going to give some testimonial on this. Selena Rodriguez is a former fellow here at Stanford. She is a journalist and also a participant who has had a lumbar puncture done in the past. So she's going to come up and talk to us about her firsthand experience with this procedure.
And feel free to write your questions on the index cards for Q&A at the end as well.
And I would add, she's an award-winning journalist. She's just, you know, her list of awards is just really incredible. I don't know if you're still with CNN, but
I did. Now I'm independent.
so from CNN to true independence, and she's been a friend of the center, a volunteer, an advocate for her community, and we're really grateful that you're here
Thank you.
to speak about how things went.
Thank you. Thank you so much. Thank you. Yes, I'm a former Stanford fellow and journalism that really changed my life. I'm originally from Guadalajara, where the mariachis come from. You all know the mariachi music. And in fact, I was supposed to be at a concert with the San Francisco Symphony Orchestra celebrating Day of the Dead. You have seen all these Catrinas and skeletons around. It's a beautiful tradition, the most well-known Mexican tradition in the whole world. But I'm so happy that I made a decision to be here with you, to get educated, because for me, being a journalist means a tremendous responsibility to inform my audience. And my dad had dementia, so that really hit me. And I've been trying to serve my community through Alzheimer's Association. That's how I met Dr. Nusha Askarin, and then we had the opportunity to do a promotional campaign about the ADRC Center in the Latino community, and there's so much work to do and so much education that I need to get as a journalist. And making my participation short, because I can talk and talk and talk for hours. I'm a participant. When they asked me if we could do this promotional campaign in radio, I do radio and I did television and printed media, and journalism is my passion, and serving the Latino community has been my passion here. I said yes, but I would love to be a participant, so I'll know what the testing is about. The community trusts me a lot after 36 years of journalism here in the States, in the Bay Area, and in CNN, and 43 altogether, considering 8 years before in Mexico. And it has been an amazing experience to be part of ADRC as a participant also. And when they asked me about LP, I said, yes, but then I said, oh, wait a moment, because I heard about paralyzing and some side effects, but I said, no, trust them. You are in excellent hands. And if I'm going to talk to my community about this, I have to go through the procedure. And let me tell you that it didn't hurt. Dr. Taylor was saying that some of the participants said, oh, are you done? Something very similar happened to me. I had no back pain, and I suffer lower back pain when I was younger. I'm 67, and I had much more pain a few weeks ago when I was very brave, and I said, oh, my house is so dusty, let me vacuum. So I was vacuuming under the some furniture and all of that. Well, 2 days later, believe me, I had a lot of pain. So this is a procedure that is amazing with all this liquid on the spinal cord. And it's just fascinating how each part of our body is related to the brain and the whole, our whole health. And we plan to do more. I have done 3 videos with ADRC in Spanish for the Latino community, so they will know about ADRC, what is the main objective and why Latinos have one of the highest incidence of Alzheimer's. And that's why I'm a restless spokesperson about this in my community. And we plan to do more videos. We need more education. And like I said, my community trusts me a lot when I'm a public events or so, members of the community come and say, hey, you came last night. I was at a community event and one of the people there said, you came to my house when 20 more years ago in the South Bay, and you did a story with my sister who had cancer, and you really made a big impact on her. And that's beautiful because I can relate to my community. They trust me, and if I have gone through all this testing, I can tell them that we need to participate more in this kind of testing and research. And this is for everybody, not only Latinos, but all of us. We plan to do more videos, on the LP procedure. And like I said, there was no pain. It was very well done, no bruising, nothing. And I will encourage you to participate on that. We need more volunteers in not only Latinos, but also Caucasian, so we can spread the word out, and it will be great. I got friends that I want to talk to who told me, you're crazy. What are you doing that? You may get paralyzed and all this misinformation. The key is to get informed. Events like this are crucial. And whoever would like to volunteer, you can contact Nusha, but I would really encourage you to do that. It's fascinating. And just to close this, because I can talk for hours, I get all excited. I consider myself very lucky that I had the opportunity to first come to Stanford in 85, 86. That changed my life and has impacted the Latino community here. Coming to these kind of events give us information that we need. We are spokesperson. Each one of you are a spokesperson in your community, in your family, with your friends, because we can spread the word. And to ask more people, we all need to participate. These are very challenging times. As a journalist, I still cry with some of the videos and the awful news that we're getting, especially now. And now more than ever, we need to stand together, to hold hands together, and to fight, in a good sense, for a much better world. So our descendants, the new generations, hopefully will have a better world. And thank you, Nusha. Thank you, Victor, for having me here as part of this. And so much work to do. I'm 67, and some of my colleagues back home in Mexico say, when are you going to retire? Or some of my colleagues here, and I said, what do you mean retiring when there's so much to do? So thank you for giving me the opportunity to be here to get educated. I still need to learn much more. And I hope we can get more volunteers. Believe me, it doesn't hurt. It's amazing. Don't vacuum your home. Okay, don't do that. Thank you so much.
Muchísimas gracias, Selena. So to dovetail on what she's saying about volunteers, we are asking for an English-speaking volunteer and a Spanish-speaking volunteer so we can get this LP video done as soon as possible. So if you are interested, please contact me. You can just email the ADRC email, since I get those, and I'll be happy to follow up with you. Thank you all so very much. And again, Selena and Ryan, thank you.
And you will not have an anonymous LPer. You'll have Dr. Taylor, I think. We thought, yes. And we also thought he'd have the best video present. Well, so, and you've also heard it. Now you can tell everyone, you know yourself, that having an LP is more fun than vacuuming. So you're
going to introduce Patricia.
Okay, so thank you. So this dovetails also on our next speaker, who is Dr. Patricia. I'm blocking. You're not Patricia. Patricia Rodriguez Espinosa, who is our colleague here at Stanford. She was recruited into the Alzheimer's Center a couple of years ago. She is not a neurologist. She's even better. She's a psychologist and epidemiologist who has a lot of community experience and doing outreach in different areas. And I'm just so happy that she's joined us and sort of working within this space, within the dementia, neurodegenerative realm of doing things. She heads up a group committee on justice, equity, diversity, inclusion, JEDI, and she will tell us a little bit about her work and where it's going.
Thank you, Victor. Good afternoon, everyone. Lovely to see you all. Lovely to be here with all of you. Actually, know Selena from another study, so it's lovely to see you, Selena. Let's see. Victor mentioned that I'll be telling you a little bit about a new program that we have within the ADRC, a program that is a little bit different. We're a little bit like an infrastructure support group for our ADRC community, for our faculty, our trainees, and our staff to make sure that they feel supportive and can do their best work, and that we're creating more resources for them. Here's a little picture of some of our members. This is our second year that we have had this program, so we're growing. These are some of our members, but we have new members since, and we have made sure that there's members from all of the various cores involved in our program, so we get a variety of perspective, and we're here of the needs of our ADRC community. Let me tell you a little bit about our mission and our goals. We have a few of them. One of them is to make sure that we foster research and research questions that might be a little bit different. We want to ensure that the science and that any advancements that we help to contribute to can also contribute to the betterment of all cultures, of all individuals, and that everyone can be a recipient of those benefits. So we make sure that there's questions, especially related to equity, and research questions related to equity, to access within this work. We also do a lot of educational and training programs, especially for our ADRC community, for some of our new members, some of our staff, trainees that might be coming from other departments. And I'll tell you a little bit about some examples in a second. But also very interested that we are a center in which everyone feels respected, feels like there's a culture of belonging and inclusivity here at the center as well. And finally, making sure that we also have representation among our workforce, in particular from various cultures, from underrepresented groups, and that this is true across all the way from our scientists, our faculty, to our staff and trainees. I highlight a few of the activities that we have had. For example, in the past year, we have created a speaker series within our goal of diversifying the research, making sure that we're asking a variety of research questions. We have brought speakers from other ADRCs, national experts in other areas to tell us about some interesting work that they might be doing at their center. This can increase our collaborations, but also ensure that our scientists are also thinking about novel and new ideas. We also oftentimes get requests from other scientists across the country for some collaborations, for some other center grants, and we're able to provide letters of support and say that we can support some of those efforts. One of those that we did this past year around imaging work got awarded, so we'll be engaging with them soon. And also to promote different research, we were able to get a little bit of funding and release some projects that were asking specific questions about equity, access, and inclusion. And we funded 2 projects this year. So one of them will be looking at adults with intellectual disability, a community that oftentimes has a lot of needs and there's not a lot of research and support for them. And another one is a work around palliative care that will be done in safety nets here around the Bay Area. So we're very excited. We got a lot of applications. We hope that this is something that we can continue to support in the future. Related to the increasing the representation of our workforce, of our scientists, we have been involved in a variety of initiatives. We always have included some new language, for example, when we seek new proposals, when we have new positions that are asking for diverse applicants. We have been engaged in interviews for new faculty. When there's a new open position, we're happy to meet with them, also tell them about the ADRC and the work that they can do here. And we have also started to attend some national meetings in this area to bring that knowledge back to the ADRC. And we have also been engaging in more education and training. Some of our trainees have done some amazing work and have brought some of that expertise here to the ADRC. There's also a lot of resources, both here within our Stanford community, but also within the Bay Area. So we have compiled a list of trainings and resources. We have those on our webpage. And we also promote it so our workforce, our scientists here, our trainees and staff are aware and can attend some of those. And we intend to do, we've been planning other trainings with a variety of other cores. And we plan to continue to do this in the future. I mentioned we are a newer group, so we're trying to integrate more with it. The various groups here within the ADRC will be doing that more. You'll be hearing in a second from our fantastic new community advisory board, so we'll be engaging more with them in the future as well. And I think I'll stop there. Thank you.
What are the topics for the speaker that you mentioned?
Absolutely.
February and March.
Yes. So one of them, for example, Dr. Amy Kine, she works in the Madison, Wisconsin ADRC and is doing some incredible work around neighborhood conditions. So they're following people, they're able to get addresses from individuals and know where somebody has lived all of their life. And they're able to build a little bit of a trajectory of variety of exposures that you might have had throughout your life, given where you have lived. So they'll be building some of those trajectories and looking at impacts on dementia and brain aging. We're also engaging with them in that project as well. So probably somewhat soon you'll see an opportunity to take a survey that will be asking you about your addresses, for example, is to be part of that study. Yeah, great question. Are there other questions for Dr. Rodriguez? No, we have an interactive section next, so I'm happy to give a little bit of time to that group.
Or a final question maybe for Selena Rodriguez or for Dr. Taylor.
Yes, please.
Question for somebody.
Is the Black community represented in any of your SADRC participants?
Absolutely.
What percentage?
I'm wondering if there's anyone for the data core that knows the percent. I want to say it's.
So the answer is small.
Yeah.
Not nearly as high as it should be.
5%. We do. Yeah. We do. It's a little hard because there's not a large population here around the Bay Area or close to Stanford, but we absolutely do. Absolutely. Yes. But we're competing with UCSF, for example, because it'll be a lot closer to UCSF than to us to come in person. So we're mindful of that, but it's absolutely a priority population. Yeah.
I wouldn't use the word compete, but because we're really fortunate that we.
Sure.
No, I think we do a wonderful job here at Stanford. I think UCSF has good people working in the same space, and they do a wonderful job as well. We want to keep everyone here, but that's not competition. It's just, I don't know.
I should know how to turn the microphone on, right? After all these years. Trying to get a community involved, I can speak from my own experience as a Latino, it takes a long time. It's a matter of trust. It's a matter of knowing the community, and it is not only translating. I remember when I started as a journalist 43 years ago, it was the big news was AIDS at that time. And it's not only translating the information, it's cultural, it's knowing the culture, it's knowing how to get close to the community. And it takes a lot of time and effort. And I know we should be doing more. Media plays a very important role in this game, and unfortunately we get to see a lot of sensationalism. We make news, Latino make news only when there's something bad or a problem with the community. We say, yes, we got high numbers of Alzheimer's among Latinos, but we need much more education. We need to get closer. So it's not an easy task. It's not an excuse. It takes time. It gets to know the culture, how to approach them. So it's a long process, but the main thing is to start doing something about it. So it takes a lot of time. Great to see you again. Not knowing you on Zoom during the pandemic times, but to see you in person. Thank you.
Thank you, Selena. Actually, I'm going to invite you, Selena and Ryan, to come up as well. We're in our Q&A session right now. So if there are any additional questions that we're going to take for the afternoon before we do the Community Advisory Board and the breakout sessions, this is your last chance, everyone. If you have any questions, please come up, Ryan, and Selena as well, and Patricia. And I'll give you a mic.
There you go.
Are there big racial differences in the expression of Alzheimer's disease, white, Chinese, and other races?
So yeah, so there are some differences that break down in communities. Obviously, race is not a biological entity, but depending on where people are from geographically, different cultural practices, and then different communities tend to accumulate a little bit of genetic difference that can predispose risk for certain diseases like Alzheimer's disease. So we do see some variability that breaks down along those lines.
But it's not actually an essential part of, let's say, its diagnosis of certain diseases or like.
I would say that race does not factor into clinical suspicion kind of in a routine fashion in clinic. Yeah, when diagnosing Alzheimer's disease.
Maybe I'll add a little bit that we see some disparities in individuals from minoritized backgrounds getting diagnosed much later, because oftentimes access is an issue that is not equal across. So we see people who get diagnosed much later, who oftentimes don't have access to specialty care. So for example, for Latinos, dementia is often managing primary care. So we see some disparities in what happens when you get the diagnosis and even after, the quality of care that you might receive afterwards. And risk factors as well, when we think about risk factors like having cardiovascular conditions, etc. We see more of those in some of those communities as well.
Yeah. So a lot of it is a lot of the difference we see amongst groups like that has to do with access to resources and general health disparities that exist in those communities for historic and economic reasons.
Yes.
Even in the Latino community, there are different levels of the society. Some who are very well educated, who got a good job, and we see a lot of also agriculture workers that don't have access to health services, and the lack of education. That's on many of my reports, I emphasize on symptoms, on very, very basic education on Alzheimer's or the brain diseases. And so it's a very diverse community, even among just Latinos. So considering other ethnic groups and the cultural differences and all of that, it's the disease, I assume you guys are the experts, gets specific angles on each ethnic group.
All right, we have a question for Dr. Rodriguez. Are research centers collaborating across different countries?
I am not familiar across different countries. In the US, we have about 37 now ADRCs, and we do try to collaborate. We share data, we share scientific advances, resources. We go to conferences a couple of years together to establish some of those practices together. I don't know, Victor, if you're aware of collaborations with internationally.
No, I agree with your answer in general. Sometimes cross-national collaborations are difficult because of some of the data sharing requirements. But that said, a lot of scientists working within this Alzheimer's Center and other Alzheimer's disease centers have international collaborations, are working in conjoint projects. I collaborate with colleagues in Denmark, for example, but there are a lot of other laboratory-based examples. I know Dr. Shula this morning has collaborations in Europe as well. And so in that sense, there are a lot of international collaborations, but not more formalized within this specific or other specific ADRCs as far as I know.
I just thought of one example, the group that I mentioned from Wisconsin that's looking at life history trajectories for neighborhood level exposures and factors. They have a collaboration in Mexico as well, especially looking at people who might be immigrants who are now in the US and trying to develop those trajectories for the time that they were in Mexico.
Yeah, in fact, doctor, you mentioned her name this morning, Dr. That she short hair. Yes. She was telling me that she goes to Mexico and she does some research with the families, with the relatives of some of the migrant workers who have been here. And but this has been related to Parkinson's disease. And she is a student from Peru. She does studies in Peru too, is coming to her office this Monday, and she will be here for a month. So there's starting to be some collaboration that I think is crucial, especially in a country like the United States, especially in a state like California with this diversity of different ethnic groups. This becomes fascinating.
I would like to emphasize within the US, we have a very well-established way of collaborating within the other ADRCs. We have a national coordinating center that we share data with, so scientists can look at data from all of the ADRCs, and they're growing as well. I think when I joined this ADRC, there were about 33 across the US, and now there's 37.
Well, technically, there's still 33. There are 4 exploratory centers. The expectation is on the next round of funding by the National Institutes of Health, if these exploratory centers have done a good job in the past couple of years, they will be elevated to sort of a regular status. The other small example I was going to give, I mentioned at the very beginning, something called the Asian Cohort for Alzheimer's Disease. This is a multi-site study. We've just received funding here at Stanford to be part of that. This is a consortium of that involves Canada as well as the US for looking for Asian recruitment and looking at Alzheimer's genetics and looking at other kinds of risk and exposure factors that might differ between Asian populations and other populations. So that would be one other small example.
So when you say Asian, that's a very diverse group.
Well, and so is Latino, Hispanic. And the answer, of course, that's very true. Here the impetus is on East Asian rather than South Asian for the time being. That doesn't mean that there isn't explicit interest in looking at broader populations, and even saying East versus South, of course, really glosses over a lot of important differences among different populations within those groups. But for the ACAD, the Asian Cohort for Alzheimer's Disease, given limitations in funding, given sort of a primary emphasis on genetics, they wanted to sort of look at a more homogeneous part of the larger Asian group. And so the initial emphasis there is on East Asian with an eye to branching out to South Asian soon.
I have another question now, elevating that to, is Alzheimer's dementia addressed in the World Health Organization at all, to your knowledge? Kind of a broad question.
Is Alzheimer's a disease addressed by the World Health Organization?
Yes.
Yeah.
Short answer.
Yes, it is. I'm not.
No, I mean, it's recognized as an important international disease.
Yeah.
With different manifestations, different prevalences in different parts of the world. There is a great deal of interest in looking at underdeveloped parts of the world and how dementia, how Alzheimer's disease is being manifest there. And so it's a good question. This isn't something that America's discovered and no one else has. This is something that's on the agenda of a lot of people, and appropriately so.
Thank you. And to dovetail on the World Health Organization, we have a COVID-related question, and that is asking if we are collecting COVID data from our participants, and if so, has there been any impact or influences on our studies in particular that we have seen?
One microphone is enough, thank you. So the question has to do with COVID, and that's such, there's no way of putting into words how important this pandemic has been to health, in the United States and throughout the world, and what the known manifestations are and what the unknown manifestations are. Long COVID appears to affect the brain. There have been some studies looking at the incidence of dementia in people who have had COVID, and some suggestions that there's an increased rate. There's some theories why, if this is true, and I suspect it probably is, why it might be true. This center and other ADRCs collected information on COVID exposure early on during the pandemic. There's a large group here at Stanford who's looking at long COVID and has developed a cohort of that. I would like to say that we're integrated with that group, but really right now we're working separately. But there are a lot of people looking at this issue. And I think some of the other ADRCs, they have been able to look at this more directly within the research that they're doing.
Yes, okay. Thank you, Dr. Henderson, for that. And this is also open to all speakers. In terms of new volunteers for the ADRC, are you particularly looking for people that have Alzheimer's disease or other neurological diseases in their ancestry?
I think this is a me question again. So it's the different ADRCs look at this a little bit differently. The overall mandate, of course, is looking at Alzheimer's disease. And the NIAs recognize that there are other disorders that fall within the space, sometimes overlapping, sometimes hard to distinguish. And so the mandate is broader. It's ADRD, with the ADRD means Alzheimer's disease-related dementia. A lot of us, including myself, don't like that terminology because it implies that other dementias are in some ways related to Alzheimer's disease, and some aren't. But all these, I think, fall within the purview of what an ADRC might look at. Here, we've decided early on that given expertise here and interest in the scientific rationale, the focus would be primarily on healthy aging. It would be on Alzheimer's disease, including early manifestations that might be apparent with mild cognitive impairment. And it would be on Parkinson's disease without cognitive impairment, And for people with Parkinson's-like pathology who have dementia with Lewy bodies, looking at that spectrum of cognitive impairment. I think in terms of resources, we really don't have the resources to expand widely into other kinds of cognitive disorders, although we're cognizant they occur, and we know very well that there's a lot of overlap in the renewal application. We want to look more carefully at vascular factors than we've done before. Does that answer the question, or? I think, there's always this tension. So those of us who wear epidemiology hats from time to time like things to be as representative as possible. And so by pre-selecting for ancestry means that it becomes, in that sense, less representative. But we don't have the resources for a large population-based cohort. I wish that we did. And so it makes good sense to try to look at people who are going to be particularly vulnerable, particularly at risk, whether it's from genetics or other causes. Right now, given our resources, we're focusing a little bit more on people who have some cognitive impairment or motor impairment associated with Parkinson's than people who are completely healthy. But, we're certainly open to all comers, and having particular risk for whatever reason, I think would add to the research value of what people are able to contribute to finding good answers.
Can you all please join me in thanking our speakers one more time? Thank you. So Dr. Henderson, would you like to introduce Dr. Goldman Rosas?
Is she here? I feel very honored today. I have such good colleagues that I work with, and I mean that just so sincerely. And one of those is Dr. Lisa Goldman Rosas, who has fairly recently joined the Alzheimer's Disease Research Center. She comes from a background focused on community engagement, epidemiology, looking at a number of different communities, but particularly looking also at Latino, Latinx communities. And she is here. One of the important things she's brought is an engaged community advisory board that she's recruited and sort of oriented somewhat as best she could as to what we're doing, and is here to tell us more about her CAB, to introduce members who are able to make it here today, and then to lead us in sort of a group discussion.
Thank you so much, Dr. Henderson. It is a real pleasure to be here today. So I have the honor of being a faculty member with Dr. Henderson in the Department of Epidemiology. I'm an epidemiologist by training, and along the way in getting my training and doing research in epidemiology, mostly in the areas of diabetes and cardiovascular disease, I learned a lot about how to engage diverse communities in various areas of research. And ever since I joined Stanford, I think now about 5 years ago, Dr. Henderson had been trying to get me involved with the Alzheimer's Disease Research Center. And among my many responsibilities related to other centers and community engagement around campus, I was really motivated by our community members who had an interest in preventing and better managing Alzheimer's disease to get involved in this really important research center. And like I tell all of my research teams and in my own research, participants are the single most important aspect of what we do. And we value you so much, and for all of the time that you spend with us. And of course, for today, for spending time with us to learn more about our research center. In community engagement, I think probably the single most important skill is not talking to people, which is what people often think, but it's actually listening. So today we're going to spend some time listening to you, and we're going to get up and move around and get into groups in just a second. And so I hope you start thinking about that while I tell you a little bit about what we're going to do. Yep, there'll be a break.
Definitely. Okay, so I'm just going to introduce our team and our community advisory board. And then I'm going to tell you a little bit about this breakout session that we're going to do. That means we're going to break out into small groups and talk together. And then we're going to report back and learn from each other. So first, I'm going to introduce my team and what we do with the Alzheimer's Disease Research Center. So our team is called the Outreach, Recruitment, Engagement Core. And so they call us the OREC team because there always has to be some alphabet soup of some sort of acronym. So we get our own acronym also. And you'll see a familiar face up there. So Patricia, who just talked with you, who leads the JEDI team, is also a key leader in our Outreach, Recruitment, Engagement Core. And we also have Dr. Wei-Ting Chen. Can you wave? Hi. And the newest member of our team, Diana Carbajal. She just joined us. I don't want to say yesterday because I think it was Monday, but she's newer than all of you, and she made these beautiful slides, even though she's just getting to know the Alzheimer's Disease Research Center. We're so glad she's here today. So let me just tell you very briefly what it is that our team does. So first of all, we build partnerships. We build partnerships with community-based organizations, other agencies, and with patients and caregivers. So you could imagine some of those partners are agencies like Alzheimer's Association, but also community groups like that one of my colleagues runs that serve seniors in the community. And then our second goal is to collaborate with our community advisory board, who I'm going to introduce you to in just a minute, to devise recruitment strategies and retention strategies to ensure that we have a really representative and diverse patient population that participates in the Alzheimer's Disease Research Center. We also work on developing educational materials and sending those out. And then we also collaborate with all the other cores, particularly the training cores and the clinical cores, to support them in building community partnerships that help with the research as well. Okay, so now I'm going to embarrass our community advisory board members who came with us today. So if you're a community advisory board member, could you stand up? So Sandy, Ben, Kirk, Leslie, and Amy. Yeah, great. So thank you everybody for coming. And I have, and Alyssa, I have all of your names listed here. We are so thankful to our community advisory board members. And you may be wondering, what does a community advisory board do? Well, each of the community advisory board members plays a really important role in our center by providing advice and guidance on the type of research we do, the outreach that we do, and all of our community engagement activities. And they represent themselves and their own experience, but they also represent the communities that they serve. So some of our participants in the community advisory board work in agencies and community-based organizations that serve older adults. And so they also bring that experience to the table, which is really important. And so we are so thankful for the work that they do. All right. Good. So this is a description of what we've been working on in our community advisory board over the last year. We started almost a year ago. I think January 10th was our first meeting last year. So we're coming up on a year. So they offer direction around services, education, and research priorities for our center. They help us build community partnerships. They advise us on how to recruit underrepresented communities into our ADRC. And they kind of act as ambassadors out in the community for our center. Okay, so what we're going to do is break into 5 groups. So I'm going to ask you to count off in 5s in just a second. And I do this with my students all the time, but we're a little bit of a bigger room, so it's going to take a minute, but that's okay. I think we need the break. So we weren't sure if everyone knew they had a number on their on their name tag. Excellent. Okay. If you have a number on your name tag, can you raise your hand with that number? 1, 2. Do I have enough of everything? Okay. Okay. I think everybody pretty much has a number. If you don't have a number on your name tag, I would like you to find a group that has fewer people. Yeah, okay. So we'll kind of work it out. We can help shift people around. So no group is too big and no group is little. So we're going to break into these 5 groups, and then one of our community advisory board members has a list of questions that I'm going to show you in a minute. And they're going to ask you those questions together. We're going to have a group discussion. You'll also have a note taker, which is going to be me. For example, I'm going to work with Ting. I'll be a note taker. Patricia will be a note taker. Diana, Wei Ting, somebody else. Anyway.
Hillary.
Okay, so we have note takers from the ADRC. We're going to have our discussion, and then one of the people in your group is going to be responsible for reporting back, and we're going to develop kind of some learnings from that. So if you're somebody who's usually really quiet in groups, I hope you'll feel really comfortable to speak up, because we really do want to hear from you. And if you're one of those people that likes to talk a lot, maybe try and give some space to those other folks who have a harder time talking. So we call that give space, take space, right? Okay, so let me tell you what we're going to talk about. First, well, I'll give you the overall idea that we're going to talk about. We're going to talk about recruitment and retention of participants in our research studies. So we've had a number of successes in recruitment, like all of you are here today. And our teams here at the ADRC have worked incredibly hard to recruit participants, make sure that you understand all the procedures that are involved in participating. And we have been very successful at retaining those participants year on and year on, right? One of the challenges has been recruiting underrepresented participants. And actually, the National Institutes of Health would like us to focus specifically on Hispanic, Latino, Latinx participants, because it's a very large group here in our area. And we've already talked about that today. That doesn't mean we're not interested in other groups. We, of course, are, but that was a challenge that they gave us. And they would currently like us to double it. So we want to hear from you what ideas we have. So first, we're going to ask you about your motivation, your own personal motivation in joining the ADRC. And then we're going to ask you what made you stay participating here in the ADRC. And then we're going to ask you to think about what barriers people who aren't here today, who haven't participated yet, might face, and what kinds of solutions can we come up with that might overcome those barriers. Now, we're interested in both the perspective of participants, but also people who may be caregivers of participants. And that's a topic that our Community Advisory Board is really interested in. Okay, any questions about the activity? Maybe the timeline would help so you know how much time we have. There we go. So what time is it now? Oh, we are just perfect timing. Okay, so we're going to spend about a half an hour in the groups, and that means about 5 to 7 minutes a question. So we'll be kind of moving along, and then we'll ask you to report back. So let's see, so we'll stand up and move to our tables. That'll give us a little chance to stretch our legs. So I'm going to have group 1 over here. Don't get up yet. Hold on. Group 1 over here. Group 2 in the back. Group 3 here in the middle, and then 4 and 5, okay? And if you don't have a number on your name tag, pause for a minute and assess to see which group needs more people, okay? Too much coffee in mine. Too much coffee in my coffee.
So while you're doing that, let me be defensive in a small way, and that is the percentages that Dr. Gomez-Rosas gave, I think, are pretty much correct, but the number of people that we have enrolled in the Alzheimer's Center now is around 400 since the beginning in 2015, maybe 500 or so originally enrolled. Some of those people have died, a few have moved away or dropped out. So the numbers are a little bit higher than what you saw. There, I'm done.
We want to be sure to capture it. Okay, I'm gonna start with, is this group 2 in the back? Okay, I'm gonna start with group 2. I'm gonna bring the microphone to you. Okay, who's gonna report back for group 2? Rick? All right.
I'm Rick. I'm here with the study because my wife's got cognitive decline and had several sessions with Dr. Fleming. And my things we discussed here is how to get this information about what we're doing here out to the public. You know, we came up with several ideas like putting some 1 minute segment on the radio or public service, or going to farmers markets or places where seniors hang out, for people that need that kind of information, so we can the public can understand what we're doing. And another issue that we have, in my personal case, is just finding a caregiver that could take some relief for me when I need to take some time off and qualify to incorporate it into my life as a third person. And how do you make sure this person does the things that you're doing as a caregiver in your own house? You know, it's very difficult. There's not a roadmap out there, you know, and I have limited, you know, like I've just said, we have so many heartbeats left, and I want to make the best experience for myself and for my wife. And just figuring that thing out, it's a real puzzle. It's very hard. You know, this is pretty much what I have. I can't even remember anything else that I covered, but thank you, Leslie. She did a nice job.
Thank you so much, Group 2. Thank you, Rick, for reporting. Do we have a reporter for Group 1? Is it you, Sandy? Okay.
Well, we had an excellent group, lots and lots of very good ideas. I think the one thing that motivated people the most to participate was this idea of lifelong learning, and that the people around the table wanted to learn and wanted to contribute to the experience of others, to contribute back to their community. One of the things that seemed to be important was using trusted community partners to help to recruit participants for studies. An idea was to have more written materials in doctor's office, and maybe doing some of the testing at senior centers, that kind of thing. Definitely making the experience to participate easier in terms of transportation. A lot of people have transportation challenges. We had a comment about the fact that this building is not very well labeled. The number is on one side of the building and people couldn't really find it. So how do you make the experience easier? And one of the solutions might be to do more of the testing in people's homes, or again, this idea of doing it in a senior center. And then some of the tests, it seems like they're very educationally dependent. So counting backwards from 107s, I mean, you struggled with math, that would be a challenge to many people, let alone people who are facing issues with cognitive decline. So, and if you're trying to do it in a language that isn't yours, that makes it even more difficult. And then, so I guess that was the last thing, making tests culturally and linguistically appropriate. Is there anything else? Okay. Yes, and that participating in a study makes you empowered instead of feeling victimized. Good job.
Excellent job, Group 1. Round of applause. Okay, I'm coming back to Group 4-5. I'm leaving my group to the last. Maybe just like get my steps in. Okay, who's going to report for this group? You, Wei Ting? Like Nusha running around. So do you want to start? Do you want me to start? Okay, all right. So what motivated people to participate in research? I think that family history, that personal connection was really important to the participants in our group. And we also learned that they got interest because they actually saw information about the center and opportunity to participate. So that's a really good reminder that we actually need to be actively putting information out there, talking with potentially education sessions with general practitioners, as well as other healthcare providers who can help us get the word out about research participation opportunities. Do you want to talk more next?
Yeah, it also sounds like the participants like knowing how their data is. It sounds like people like knowing how their data is being used, and they also like knowing what we're looking for so they can tell their friends and family members appropriately. Like some people expressed not necessarily knowing that we were trying to recruit more underrepresented people. So now that they know that, they can spread the word properly.
And Jeannie also reminded us that it's really important to reach out to our current participants to help them provide testimonials and encourage others to participate. I don't think that's something we've talked about yet. Holding events, regular events updates like today. We heard a lot of kudos about the short talks and really, really good slides. So thank you for that feedback, and use these opportunities to just help people get better understanding of what's happening at Stanford. We also talked about retention ideas. Again, that personal connection and commitment is what keeps a lot of people coming back. But also, the research coordinators are amazing about accommodating participants' schedule. That flexibility is really important, and that's really helpful. And then creating more opportunities to develop relationship with center staff, researchers. I think that's also something that's a retention and engagement strategy. And having events with good food. Where's Nusha? Thank you. It's really important. Yeah, I'll pass this back over to Hillary.
Yeah, and we just got some really good specific examples for ways we can recruit, like through radio. Sorry. Okay. Through radio stations or specific Alzheimer's disease groups, community centers. So thank you everyone for the very specific recommendations.
And I think that's it from us.
All right, round of applause for group 4-5. Excellent. Okay, my group, I think, is very shy about having a reporter to share back. Who wants to do it? It can't be me, I'm already talking. Ting, you want to do it? Yeah.
I think our group actually very diverse. From people from Fremont, Dublin, and various places. So distance seems to be not a problem. One particular, just like the other group was saying, the trust of the system is very important, especially from somebody from Redwood City mentioning it. One of the idea we start floating around, because it seems that our group represents very highly educated, very privileged group. So what we raised the question is, if you are going to take away the participation money, but actually use that to have higher participation reward for the underprivileged community, would that help? I think people should think about it a little bit, because in order to have the underprivileged community to get more involved, somehow you have to sort of pay them enough so that they can take the time and so on and so forth to join the research. I think people in general from our group is really interested in learning more, because again, it's purely because it's highly educated participants. So I don't know what else to add.
All right, round of applause for group 3. Great, so maybe I'll just summarize a little bit what I think we heard across the different groups, both in my group, but also listening to all the groups. I think a lot of the motivation for participating came from wanting to contribute to knowing more about this condition for yourself. And most importantly, what I heard was for family members, right? So that seems like a motivation that we can amplify for other communities, because it will be relevant for all communities. Another thing we heard a lot about was a motivation to participate, to just learn more about the condition, right? And it sounds like through events like this, and even through your participation, that a lot of people are learning about the different forms of dementia and Alzheimer's through participating and interacting with the study staff and clinicians. I think you all came up with some really important solutions that I heard in terms of making the study more accessible to more people in our community, around addressing issues of awareness and access and trust, things that we can do to really engage with other communities so that we can make the benefits of this study available to all. So I want to give everybody a huge round of applause for the work you did today. And I want you to know, if you have other ideas, you now know a community advisory board member for the Alzheimer's Disease Research Center. And remember, I said they're ambassadors, so you can talk to them and let them know the ideas you have. You can also speak to our team. I'm Lisa, Wei Ting, Patricia is somewhere, and Diana is right there. We're always open to hearing your ideas about how to make participation better for you and also more accessible to others. So I think I'm going to pass it back to you, Dr. Henderson, to close out, right? Oh, and Nusha has something too. Thank you.
Oh, thanks so much. Thank you, everyone. And I won't close out. I think Nusha will do that on our behalf. And I also want to sort of emphasize that, this is a center that's trying to get to, find out answers to neurodegenerative diseases that affect cognition and aging. And so even though we're an Alzheimer's disease research center, and there's maybe a lot of stigma associated with that within the Parkinson's community, in fact, the title is a misnomer because both of those are really of important interest to us. And so I hope that message goes back to the Parkinson's disease community as well. All right.
So thank you again, everybody. I think all of the ADRC would now like to give all of our participants a big round of applause. We greatly appreciate your spirit of generosity, your volunteerism, your involvement and engagement, and furthermore, your ambassadorship going forward into the community, trying to spread the word, educate people, and hopefully get them involved with the ADRC and other opportunities. In closing today, first of all, again, I want to thank everybody who's been involved in organizing the event. So I have a heartfelt thanks to Alyssa. Wave your hand. Thank you. She's actually going to be transitioning to another position soon. So I'm deeply grateful for all of your engagement and involvement with us. And I'll be sad to see you go. Also, Jamie Kramer. Jamie, turn around a moment. Thank you so much, Jamie. Greatly appreciate your help. Christina Weiss-Corey, who, as you all know, she does so many things and we greatly appreciate her. And helping her today were Hannah, yay, Nicole, she might be outside, Nora, Leah, there you are, yay, thank you, yes, Samantha, yes, and Hillary, thank you all so very much. Sorry if I was remiss in anyone. And then lastly, we ask that you please remember to fill out the survey in the link in the bottom of your agenda. I had emailed you all the agenda. Please go and connect through that link and give us your feedback. We'd greatly appreciate it. And then last but not least, there's plenty of food out there. Please help yourself. Take a plate and take some food. We want you to stay healthy and well, and well nourished. So please go ahead and help yourself to some food. Thank you all again so very much. We'll look forward to the next event and we'll make it hybrid, but we want you to come in person so we can see your happy, friendly faces again. Thank you all so very much again.
And remember, you have an extra hour tomorrow to eat the food that you take home today.